Todd was extremely excited about being a part of this. He actually has a kind of interest in venture capital, meeting people, but a very potentially high-value recruit is in Oklahoma, and so Todd is flying to Oklahoma today. But we made a little video, and I'll go ahead and
themselves. And I think one of the mottos that we use with our team is team first, and we talk about that every day.
That certainly applies to building a company and building a team, I think. Todd, I know the whole Gator Nation is super excited about you being here, and maybe you could just, as we wrap up, just tell us a little bit about what you think the future of Gator basketball is going to look like.
Yeah, you know, it's an amazing place here in Gainesville. Obviously, a lot of great tradition and history, dating back to the late 90s, early 2000s with Final Four runs, what Coach Donovan did in the mid 2000s, and you know, my goal for this program is to get it back to those heights where we're competing for national championships on a yearly basis.
Yep, and to that point, Coach Golden can't be there today because he's got more important business with recruiting some new Gator stars. So, Todd, thank you very much.
Yep.
And go Gators.
Best of luck.
Yeah. So there are little black books, I think, in front of you. If you could look, one of those books has a golden ticket. We've got an autographed basketball here from Coach Todd Golden, and one lucky winner is going to take this home today. Go Gators. So, W.J. Rossi wins the golden ticket. All right, congratulations, W.J. You don't need season tickets, so I know that the basketball is something for you. All right. It's really a pleasure for me to introduce the next speaker from DEO, Department of Economic Opportunity, the state of Florida. And if you think about, you know, oftentimes people that work for government, you know, you kind of wonder, well, what's their background? How's it relevant to what their job is? Well, that's definitely not the case with Mike DiNapoli, who's a former senior vice president at Citigroup and now runs new business opportunities for the state of Florida. Mike, it's a pleasure to have you here. Thank you.
Thank you, Jackson. Thank you, Jim. Thank you for the invitation, and thank you all for being here. It's great to present and kind of showcase what we do at the state. A little bit of background about myself that, as Jim had alluded to, I worked in finance for some 20 years, working in New York for my whole career, and then moved to Florida. I was an early transplant, I think, back in 2012 when I bought a horse farm and kind of got out of the finance world for a little bit and built that business up. For good or bad, it's now a thriving business. I gave it to my daughter years ago, and after my dad told me, you know, never have inventory that eats while you sleep. It was one of those things that I learned very quickly how expensive that was. So God bless her and what she's doing. But that brought me to Tallahassee. It brought me to the state. My way of giving back, I started a business, started multiple businesses, and it was a learning and it was a challenge. But I'm glad that I did it, and I'm glad that I was able to learn from it. And taking what I've learned and the things that I've done in my past experience with Citigroup, with Solomon Smith Barney, with Smith Barney, and the whole maturation all the way through to Morgan Stanley of all companies, where my wife started out years and years ago. I wound up at the same company she was at. So it was a ride, and we moved to Florida and never looked back and love it. Moved up to Tallahassee back in 2019, started with the state, started with DEO, the Department of Economic Opportunity. It's an executive agency. Absolutely love working there. So those of you who don't know what it is, look it up. We do some amazing things, not just from what I do, but some of the other things that we do for community development, for unemployment assistance. It's an incredible agency that came together years ago back under the Scott administration back in 2012, I believe it was, 11, 12, where 3 or 4 different governors, agencies came together to create one Department of Economic Opportunity. So I'm going to go through some slides. I'm going to kind of talk a little bit and describe what we do and why we do it and how I think it's so important. I was just had the honor to be down at the Florida Chamber in Orlando, and I did a little R&D, what I call R&D, a little bit different from what you all call R&D. I did a lot of rob and duplicating of some of the information that was shared. So I'm going to share some of that. I took some notes while I was down there, and I think it's a lot of information that you all know about. You're in the business, you know about it. I kind of look at it a little bit different, and I look, how can the state help? What can we do to help? We're not trying to take over. We're not trying to jump in your sandbox. We want to know how we can help. So there's a program that I want to mention that's extremely important to me that I inherited back when I started at DEO in 2017. And it's now been reauthorized, a federal program that's been reauthorized. And I just want to touch on it because I think it's incredibly important for businesses here in the state of Florida. So clicker works or arrow works. There we go. So here's some of the numbers that I R&D'd, and I'm sure you all know this and you've all seen this before, so it shouldn't be of any surprise to you. What I found interesting, am I going too fast here? Oh boy. Does the screen not connect to the computer? It's not, it's page 3. We go back, but does it show up here? It doesn't show up there? That's okay.
Yeah.
Doesn't go back. Oh, you can't go back? Okay. Well, we can't go back. So some of those numbers that were up there were the overall market that you all know about. When I took the numbers, $995 billion, but then I look at it and see what does Florida's numbers look like? Where does Florida stand? And kind of going to show you, I'm going to walk through what's been done in the past and what we're going to do in the future and some of the things that we're going to focus on. So what I found interesting when I was down in Orlando, and this will be on one of the later slides, but I'll bring it up now, is the percentage of Florida when it comes to VC, the percentage of Florida, what it is overall in the market. And last year, I believe, through the first half of 2022, Florida had 2% of the market. I look at that and say, oh my goodness, why? What is the problem? What do we need to do to become a bigger player in VC? What do we need to do to help the market? And what UF Innovate's doing, and some of the other tech transfers and some of the others around the state, like USF and FIU and what they're doing, and even up in Tallahassee with the Jim Moran Institute and some of the things that they're doing. How can they do better? What can they do to make things better? So it's kind of interesting too, some of the numbers that I also wrote down in Florida, and it blows me away that Florida only accounting for 2%, but then when you put Boston, New York, and the Bay Area, incredible numbers. It's nearly 3/4 of the market. I think California does more in a day than Florida does all year. It just truly, I wouldn't accept that. If I were in your shoes, I wouldn't accept that either. I think we can do better. So let me just jump ahead to this slide here, which is talking about the program that we've done in the past. So back in 2007, the Florida legislature passed something called the Capital Formation Act. And it was really in response to the monies that we were getting from the federal government under a program called SSBCI, State Small Business Credit Initiative. And I'll mention that because they just reauthorized it. It was put in place to help, I thought, when it originally came into place, it was thought to help the smaller businesses try to get funding, the smaller capital investments that really needed some funding that were getting overlooked. I don't think that's the case anymore. I think with the amount of businesses that are here in Florida, over 3 million almost 3 million small businesses, over 3 million employees in those businesses, they're not being overlooked. They're not being overshadowed by the bigger small businesses. So this program that I will mention going forward is going to be a very important tool. But through our partners at the Florida Opportunity Fund, and thank you, DeepWorks, they are our current investment manager. for the Florida Opportunity Fund. There's 3 different pools of funding, and you can see where it came from different capital. The fund of funds came from legislature. Clean energy came from, I believe it was from U.S. Department of Energy, and then our Florida Venture Capital from FLVCP, which came from SSBCI 1.0. About $42 million came from that program. I think I broke it. Doesn't want to go forward. Sorry. Should I use the arrow buttons here or the clicker? Okay. So through 2021, this is right from our partners. Those of you who don't know, the Florida Opportunity Fund sits under Enterprise Florida, EFI. And these are the numbers through June of 2021 and what they've done. You know, the 4.7 in private capital leverage, I think, is tremendous. It's going to be an important factor because for us to get our full allocation going forward, we're going to need that leverage. So what are we doing now? What is DEO doing now? We are doing, and I am traveling around doing a tremendous amount of work, making everybody aware, making the industry aware of what we have. About a year ago, when I found out that Congress was going to reauthorize SSBCI, which is about a decade old, it's now in the American Rescue Plan, which is where we got our money, or getting our money. In fact, we just signed the allocation agreement yesterday, and I'll throw a number out. Those of you who don't know, but Florida is going to get about $500 million. It's the 3rd largest behind California and Texas under this program. And you'll see that on this slide here. We are going to allocate up to $100 million, potentially more, and I'll explain that too, $100 million to venture capital. So when I first heard about this, I told Secretary Eagle I thought it was important that we make a big deal about this, because if venture capital doesn't know that we're going to do venture capital in the state of Florida, they're going to go somewhere else. They're going to relocate. They're going to staff up in other cities, or the cities that get most of the funding, Boston, New York, and California. So they agreed. They thought it fit within the governor's initiative, and under his leadership, we've now put and allocated $100 million, where Texas is not even allocating a dollar of their new funding to venture capital, and I can't understand why. They're going to quickly realize that that's going to be a mistake. And I've been talking to some of our partners about it. We need to leverage this portfolio in order to get our full allocation. And I don't know how in the world Texas is going to leverage their portfolio without using venture capital, because it is a great tool. It is an amazing tool to leverage private capital or other capital when you're in your world. So this is it's going to be a multi-year initiative. The funding keeps the federal characteristic for about 9 years, and then it becomes state dollars. That's exactly what happened under 1.0. The 2017, the federal characteristic expired, and now we invest the money nearly identical to the way we stood it up back in 2012. Before I got there, but the way we stood it up in 2012. Next slide. So here's just a quick description of what it is and what they support. There's some metrics that we need to hit with inside of our allocation. I don't want to get too much in the weeds, but per U.S. Treasury, I will always have to say per U.S. Treasury, just so you can read some of those, some of these titlings and some of the requirements. There's a lot of requirements inside of Treasury's guidance and guidelines that we have to stick with. It makes it a little bit more challenging, but we can do it. Most important, but Treasury believes is our allocation, and I'll move forward in a minute. Since I can't go back, I'll go forward in a moment and show to you how they're giving us our money. Our money is going to come in 3 different draws. We just signed our agreement yesterday. Happy to say it took painfully almost 9 months to get it done. Our first draw is going to be $142 million, of which $30 million of that 142 is going to go towards venture capital. We have different hurdles that we need to meet. One of them is socially and economically disadvantaged. The other one is very small businesses. We need to hit those allocations, and it's important I mention it because if we don't, we could potentially put our entire allocation in jeopardy. We do hit those metrics, we could potentially get more funding. on the heels of other states not being able to hit those metrics. So I'm going to throw another number out, which I hope is a staggering number to you as it was to me. I think we're going to hit over $650 million of allocation to the state of Florida. So here's what I'm talking about. Here's the allocation. 488.486 million that we're getting. That's already been signed off on, that we will receive. They tell me $142 million will be in our bank account middle of next week. I hope they're right, because we are ready to get to work. We've already got about $20 million allocated. Of course, the $30 million, as soon as we award that, we did a public solicitation for another manager. Hopefully we can award that soon so they can get to work. There's also a timing issue in a lot of this. We have 3 years to get the money out. We have 3 years to get $142 million out, and we have 3 years to get 80% of that before we can go back and get more money. So more weed stuff that I don't really need to go into, but let me just pause there for one quick second while you're staring at this. One of the things that I find challenging in trying to connect public and private, we have a lot of private, public-private, P3s they call it, partnerships, Enterprise Florida, Visit Florida, Career Source, you name it. There's many of them out there that we work with, not to mention the Florida Chamber. The Florida Chamber does an amazing job, and I didn't realize how many chambers there were around the state, but in connecting all the chambers, they have what they call a scorecard, and I encourage everybody to go out and look at it. I sit back and I say, how can we help businesses here in Florida? What can we do in Florida to help businesses grow, succeed, whatever term you want to use? How do we help businesses? They have a great scorecard out there that says, if you're not listening, listen to this. They have a 2030 metric and what we're going to look like as a state in 2030. Which is not too far off. We're going to have another 3.5 million people living here in Florida. To accommodate that, we need to create, according to their score, their metrics, we need to have another 1.625 million jobs. For those of you that don't know anything about Florida and workforce housing and everything that goes around it, that's going to be a challenge. The infrastructure, what needs to happen, low-income housing that needs to happen. That's a massive support infrastructure that needs to happen. Thankfully, with the leadership of this governor who believes that we can do it, I also think we can do it. But that's gonna grow the state of Florida from the 15th largest economy in the world, which we are today, to the 10th largest economy in the world. I don't know any venture capital company that would not want to be here in Florida to capitalize on that potential growth. To me, it's just a staggering figure. The other figure I want to share with you, and this is all according to their dashboard, and you can look this all up, it's publicly available. And I'm going to steal another thing from somebody that I heard the other day, which I've already been talking about, but what's going to help the economy grow when it comes to VC? What is it? How do we build UF into this incredible tech transfer, this tech innovation center? What do we do to have that ecosystem? What is it going to take? And I've heard this before, and I'm sure it's no news to you guys and gals. Talent and capital was one that I keep hearing about. Talent is important. Of course, capital is important. When you bring in, by 2030, an additional 3.5 million people, capital's coming. You look at the metrics, and we track the metrics by people that are coming to the state of Florida and how many driver's license are given up and reissued in Florida. That's how we track all of these numbers, or they track all these numbers. But so we know that the talent is coming. We know it's here. We know it's going to continue to come. What about the capital? How is the capital going to get here? What capital is coming? And I was talking to Mark Wilson about it not too long ago, who's the president of the chamber, who said that too is coming. They are now tracking capital coming into Florida by the hour. $2.7 million in capital comes to Florida every hour. Income migration to Florida, $2.7 million every hour. That's incredible. I would love to be a VC company right now in Florida. I would love to be sitting back and saying, those people who are coming, they're going to start businesses. They're going to need investors. They're going to need people to talk to get started, to get their business going. And I think that's why it's an exciting time, one of the exciting times here in Florida. We have another facility within inside of this, which those of you who might do VC and also have any kind of lending programs, this is just another part of the program that we're going to stand up. $350 million is going to be a part of the 488, potentially more, for participation guarantee and a collateral support program. By the way, we already have $98 million in SSBCI 1.0, 42 million in venture capital, and 50 million in loan participation that we currently use. So we're just going to flip the switch when we get $144 million next week. Here's our allocation for the capital access program, $38 million. And here's VC. This is what this is why we're here. Venture capital. I think it is an incredible tool to help small businesses. It is an incredible tool to help those innovators, to help people who don't have anything but an idea, who don't have anything but a mock-up that we can invest in and that we can certainly turn into a business. One of the focuses, and on my last slide, I'll show it to you. We told Treasury on our it was an entire application process, and they liked some things, they didn't like some others, but we told them that our focus was going to be on early seed stage capital. That's where we want to focus. Another number I stole the other day, 70% of all VC is late stage investing. I had no idea. That tells me that, you know, the Googles and the Apples and the Amazons are, you know, a thing of the past where they made not millionaires but billionaires out of, you know, the initial investors. So, and also they're waiting longer to go public. They're waiting much longer to go public. Multi-billion-dollar companies now going public later in life, later in their maturity stage. So we're trying to start those initial companies. I hear some of the names of companies that have started here. Rivian was on the Space Coast. I had no idea that they were on the Space Coast. They left and got pulled away. There's Luminaire is another one that just did a SPAC recently. So there's plenty of them out there that we could have kept here. One of the things that I continue to see is companies going elsewhere, businesses going elsewhere. We used to have an incentive program, a tax incentive program, to bring big business, large businesses here. You'll hear it called corporate welfare and many other names, but we don't need that. We don't need that in the state of Florida. These companies are moving to Florida, large and small, every single day. There's roughly 1,000 people a day moving to Florida. I was just down in the Villages for the first time in about 3 years, and I was blown away the amount of people now that live there and the building that's going on down there. 1,000 people a day are moving to Florida. That's incredible. Net immigration from not just, as the governor, if you've heard him talk, it's not just New York, New Jersey, the East Coast. It's the West Coast. It's people from Washington, Oregon, California, where they used to go to Texas and Arizona. They're now coming to Florida. And I find that mind-boggling that they come cross-country to come to Florida. But I know why. We're an open state, free state, no income tax, which businesses love. We are an incredibly business-friendly state. So I know why they're coming. So here's what I was talking about. You know, we want to invest in startups. We want early startups. We do not think that it's a big risk. If it's done right, we do not think that it's a major risk. Treasury has told us they want it, they like it, we're going to do it. We're going to push early stage, development phase, idea concept, all the terms that you want, friends and family, throw all those names out there. That's where we want to be. That's where we want to push the dollars out. When I first started at DEO years ago, somebody had told me, I was telling earlier, I think Jim, that I run a program that right now we have $50 million to help those businesses that were affected by Hurricane Ian. When I started this program, when I ran that program for the first time, it was during Hurricane Irma. I started at DEO 2 weeks before Irma hit, and I got $50 million to do there. And somebody had told me that the repayment, they were asking me about the repayment, and I said, the state kind of doesn't really care whether they get any interest off of that money. They just want their money back. I mean, that's, to me as a former finance person, that's mind-boggling. You know, money has time value to it. So you don't care whether they pay it back 10 years later? No, we just want our money back. Okay, we'll get your money back. I don't want to say that's the same thing here, but, you know, I understand that, you know, the dollar has time value to it. And if it means that we've got to sit on an investment for 10 years, we're willing to do it. If we need to sit for a year and it turns into a decent return. But my goal is to take this $100 million. We need to leverage that money in order to maintain our allocation and potentially get an additional allocation, which I believe is going to be another 100 million plus. The Secretary of the Treasury, she has incredible amounts of power to put another $1 billion into every state. and every state who gets it's going to be a state, hopefully not just the blue states, it's going to be the states that are doing and hitting those metrics. We're Florida. We all know that we're at odds a lot of times with the federal government in many ways. I don't want to be that state where we just miss our bogey. We just miss that metric, and we're not going to get our allocation for a very small business or additional allocation. I want to hit those numbers, and I can't do it without you folks. I can't do it without the power of compounded, the leverage that you all offer. You'll hear from Ben, I'm sure, from DeepWorks about the work that they've been doing. They manage our current investments. That no longer is federal characteristic. Our new money, whoever wins that bid, they're going to have a challenge. They need to leverage those monies. I'm going to be leaning on them every day to leverage those dollars, because that's going to be paramount for us to get additional money. This is a lot of money. I look forward to answering any questions that you might have, or any concerns or things that we could do better. I always say I don't know what I don't know. If there's something that we're not doing right, I'd love to hear about it. If there's something we could do better, I would love to hear about it. So I just want to end on this. This is my last slide here. I think we can build this ecosystem here in Florida, this tech ecosystem. I think it begins with UF. UF is leading the charge, I think, on this with Scripps now, and I know Jackson's been down to Palm Beach a lot, going down and trying to build that up. We as a state, I know we invested in that and it failed years ago. I don't know why, but it was not a good investment for the state. I think now that it's under UF, I think it's going to be an incredible a tool to help bring businesses and just foster that ecosystem. And with all the other 12 colleges and universities around the state, I think if they work together, I'm hearing that they are, it'll work. It will definitely work. I would love to be back here in a couple of years. Maybe it's the 5th, 6th, 10th year anniversary of this, and say we are not 2% of the market, we are now 20% of the market. I think it's possible. I think there's great potential here in the state of Florida. Every day I turn around and bigger, the large VC companies, whether it be Andreessen, whoever it is, they're just adding more people. They're just bringing more people down here. I think it could happen, and I think the capital can come from Florida-based companies. I thank you. I appreciate the time. Thank you for the invitation. There's our phone number. There's our contact email. My number, if you want to call me directly, is 8966, 717-8966. I'm happy to talk to you, and thank you.
Okay, we'll get into some company presentations. This is the Fast Break Summit, but there are no breaks. It's no timeouts. So if anybody wants to get food, bathrooms are back there, just go up and do it. And now I'd like to introduce Max LeMaitre from Mattrix.
All right, welcome. I want to start by thanking UF Innovate for putting on this awesome event. I'm happy to be leading off this group of amazingly talented innovators. You know, for my money, you guys should be able to fill this stadium with the great work that you're doing. So just happy to be a part of it. I assume I'm going to have the same problems. Well, maybe not. All right. Okay, so I want to talk to you about, so first of all, I'm Max LeMaitre. I want to talk to you about Mattrix Technologies. And this is we're a bit of a rare bird in these parts. We are a hard tech startup company, which I hope that with this amazing ecosystem that we have, we'll be able to really change that and bring hardware innovation back to the US and hopefully here to Central Florida. So displays are how we interact with our digital worlds. They are literally front and center on some of the most exciting electronic products that we have today. But for as much probably unhealthy time we spend looking at these displays, we don't really think about where they come from or how they're manufactured. But you know, that's a bit strange, because I think it would be the next iPhone launch or Microsoft tablet launch would look weird without the display. It really is an integral part of those devices, the part that we see. The problem is that no flat panel displays are currently manufactured in the United States. And worse than that, over 85% are manufactured in two countries, just China and Taiwan, and that number is growing. So this is not a diverse supply chain or a robust supply chain. In fact, it's far worse than the much fretted about semiconductor supply chain that the US government has pumped hundreds of billions of dollars to help support. So it's easy to understand that a relatively minor disruption in the supply of advanced displays to the US would have incredibly problematic knock-on effects to a lot of our industries. Some of those industries include aerospace and defense, automotive, and healthcare. These are already, the display components of these industries are already very large and growing rapidly. And so they're all actually core to our national security in addition. So the overall OLED display market in the world is approaching 45 billion today, and it's expected to grow to over 100 billion by 2030. And as you can see here, none of that production is here in the US now, and there is no plan to bring production to the US going forward. And we see this as a major problem. And our hope at Mattrix is that we should be bringing some of that production in the future to the US, and we think that we can. So Mattrix will manufacture the world's most advanced displays domestically here in the US. And I understand that's a pretty ambitious goal, and you're probably wondering how we're gonna do that. So at the core, in one sentence, what Mattrix has done is invented a new way to manufacture displays that dramatically reduces the costs and simplifies the production process. So how do we do that? What we've invented is something called an organic light emitting transistor. So displays are made up of millions of little pixels that are smaller than the eye can see, and these are very complicated. They have lots of components. This device combines three of the essential components in that pixel into a single, very stable device. So the analogy here is combining the light bulb, the battery, and the dimmer switch into one highly efficient device. And it turns out that that device is completely compatible with existing materials as well as future materials and processes for manufacturing. So with this OLED device, as we call it, we can make what we think of as an ultra simple pixel circuit. So each one of those millions of pixels in your screen now only needs two components. That's in contrast to the 9 or 11 solid state electronic components that are typically required in a display. So by reducing the complexity of that circuit, we can use more of the pixel for light emission, which means we can get much brighter pixels. And then we can use simpler manufacturing technologies, which allow us to more easily make flexible or transparent displays going forward. And then with this ultra simple pixel circuit, we now have the opportunity to have a very versatile panel manufacturing paradigm, which allows us to manufacture the most advanced OLED displays using much more mature, older LCD manufacturing technology. That leads to fewer production steps, lower costs, and lower processing temperatures. So this is the core of Mattrix's technology. It's patented by a wide range of patents globally that span materials to device to implementation in products. Okay, so overall, what does this allow us to do? We believe that this is the only path to profitable display production in the United States. We can reduce the CapEx cost needs to build a factory by over $700 million, reduce the number of processing steps by more than 30%. And with those efficiencies come benefits for greener manufacturing, lower CO2 emissions, and energy use for production, as well as the elimination of trans-Pacific shipping. And finally, because of this unique pixel architecture, we can produce some really interesting next generation displays and differentiated products. We can achieve higher brightness while still maintaining the high contrast of OLED displays. We can reduce the weight by moving to very thin glass or plastic substrates, improved energy efficiency, and then most importantly, because of the simplified manufacturing process, it now makes it easier to move to really interesting products like fully transparent displays, flexible displays, or even free-form shaped displays. Okay, so I said we want to actually manufacture these things in the US. How are we going to do that? So traditional LCD manufacturing is heavily concentrated in Asia, and LCD has become a commoditized product. It's very difficult to make money producing these older generation displays. By 2030, something like 45 LCD factories will be forced to shut down. So our objective is to acquire low-cost LCD equipment that's fully depreciated at fractions on the, or pennies on the dollar, add our core IP that I talked about, this OLED, and then start producing state-of-the-art high energy efficiency displays here in the US for critical infrastructure sectors. So kind of in summary, we're combining this older LCD, simpler manufacturing process with our state-of-the-art OLED front plane technology. We're going to do this in a relatively modest size fab that allows us to have very low tooling costs. And the benefit for that is that it allows us to make truly tailored display products for those industries that I talked about. So you can imagine a premium car company having a different, unique, dash conforming display, not only in each of their cars, but also in each model year throughout. And that's something that other display makers won't be able to do. And it also allows us to have a scalable capacity so that we can ramp this with demand as we build the market here in the US. So the economics are pretty staggering. If we were to build an OLED fab using conventional technology today, it would cost at least $1.1 billion. We can reduce that to less than $300 million, which is 65% savings in the CapEx costs. And the net impact on the value of that factory project is an over 10x increase in the net present value of the project and a 6x increase in IRR. So a happy coincidence is that the US actually leads in many of the key supplier categories to the display industry, and those are going to be critical for us to be able to scale this going forward. So we will be able to partner with big companies like Corning or PPG that supply critical materials to this industry without having to go abroad. And we believe that really we are the last puzzle piece here to be able to bring display manufacturing to the US, and that's made possible by this innovative technology that we've developed. Looking forward, we want to manufacture displays here in the US for those critical markets that have supply chain constraints, but this technology generally lowers the barrier for display manufacturing in general and will allow us to export this to a wide range of countries, including Taiwan, the Quad countries, Australia, India, and Japan, and Europe, and allow us to really broaden the commercial manufacturing base for these technologies going forward and create a truly robust supply chain for these critical products. So we're still a startup founded here in Gainesville, but we have been working with a lot of really critical companies in this sector. We have partnerships with some of the largest companies in the world and several universities on the technology side. We've stepped up the technology from a little single pixel lit up in a lab to a full video, full color display prototype. Very strong IP portfolio that covers, again, everything from the core device to the application. And we've seen validation from both the scientific community and industry in the form of awards and publications in some of the most prestigious journals. So we have a really outstanding technical team. We had 3 initial founders, and we've since added a lot of key personnel to our advisory board that bring on manufacturing and execution experience that will be greatly needed to take on this challenge. And we are still, at a fairly early stage, but we are taking a very big step here in the next year or two to move to a full pilot production line, demonstrate our manufacturing, and really setting up our pre-production to be able to produce here in the US. So we're raising a Series B fund now. I won't get into too much of the details, but happy to talk to people afterwards if there's interest. But I'll just leave you with this summary. We have invented a breakthrough photonic device, essentially, that is going to enable us, we think, the only viable path to display production here in the US, allow us to supply critical markets and create some really interesting bespoke display products going forward. So thanks for your attention. I appreciate it.
Thanks, Max. Next up, it's really my pleasure to introduce Greg Sawyer. He was clearly, as you'll hear, one of our most valuable players, an amazing engineer and scientist. So, Greg, tell us about Aurita.
Thank you all very much. It's really a pleasure to be here. So, hit spacebar. There we go. So my background is I'm a mechanical engineer. I worked on the Mars rover. I love space exploration. I loved working on what I thought were the hardest problems, often around human exploration, hard materials, surface science, atomic force microscopy, things like that. I cut my teeth at NASA JPL. Today, it's a little humbling to tell you that I think the hardest problems in the world and the place that we need the most convergence between disciplines is in soft matter. It's in health. And particularly, my story is I got into this through, well, basically the hard way. It was a cancer diagnosis. It was a surprise cancer diagnosis, stage 4 metastatic cancer. And when I went back to my lab, I felt like I had zero tools to give to that problem. And it was humbling. And what I love about the University of Florida and what I love about universities in general is collaboration and the opportunities to take things from disciplines that may initially appear to be unconnected and put them together to generate new ideas, new technologies, new opportunities, and new solutions to old problems. Sometimes what is easy in one field has been a major obstacle in another. And about a year ago, we graduated the technologies out of our lab and started a company called Aurita, which we named after the jellyfish. And my colleagues here, Jack, Stephanie, and Ryan, all of us have put our effort in to basically taking the tools and the story of that lab and moving forward. So I'm lucky to be here, extremely lucky to be here, largely here through technologies that you're going to hear about with sort of a hero of mine, Duane Mitchell, working in immunotherapies. But when I got cancer and I thought about it, this is what I was looking at, okay? It was a diagnosis over here on the right. It'd probably been with me for years. The progression of that and the outcome of that was pretty bad. It had already metastasized. If we look at what was happening in medical research, it was you start with the mouse, kind of the largest mammal that most of us don't feel passionate about. And you go from a mouse study, maybe you go up to canine studies, but this is a terrible model for design and innovation, right? If we think about how we invented flight, two brothers that were bicycle enthusiasts started flying kites on a beach where there was a lot of wind, eventually take the kites and start to make gliders and start to make other structures, had to invent a propeller, had to put all these things together. The way they were able to go so fast is that they were able to have a model system that they could work with. And I felt as an engineer, what was really missing in cancer research was a model system, something other than the mouse. This little plane that you see right here, some place that we could take all that complexity and put it into one single place. If we go back 1903, cancer diagnosis, probably the best outcome is you go in and you surgically resect it. Same point in time, these guys are flying kites in Kill Devil Hills, Kitty Hawk, North Carolina. Today, we flew a helicopter on Mars a couple years ago. Today, if you've got cancer, probably the best bet is to have somebody cut it out. There's a lot of opportunity for improvement. As the field, I know that all of you are here as venture capitalists and other entrepreneurs, the field is really still what I would say fragmented. We're still trying to figure out the best way to put all of this together into these in vitro models. And I think that if you look at it, there's evidence that it really is growing. If we look at research and we see what's going on with publications and citations and how many people are working in this field, there's a tremendous amount of activity. It really is growing exponentially. But as a field, we really haven't rallied around any particular solution. So it's still extremely fragmented. If we do look at kind of the size of this, it's staggering. These are enormous financial costs that people are looking at, and these are humongous problems. So how did engineers get involved? I confess, it was not a fast start. I can assure you essentially all of the ideas I had in the first 3 or 4 years were pretty terrible. The reason is I spent most of that time feeling really guilty. I lived 90 days at a time between my diagnoses. I tried to read Nature magazine. I tried to read immunology textbooks. I tried to sit in grand rounds. I tried to think like a biologist. It was awful, and I was bad at it. It didn't really turn the corner until I decided to just think like an engineer and to be comfortable with who I am and the backgrounds that I have. And this is how engineers think about problems. We put the problem in the middle. We put the tools, the task, and the integration around the perimeter. What do I mean by that? Civil and coastal engineering, environmental engineering, electrical engineering, mechanical engineering, aerospace engineering, automotive engineering, biomedical engineering. The tools around the perimeter are the tools that we have. Solid mechanics, fluid mechanics, thermodynamics, okay? Heat transfer, robotics, all of these things are tools that we have to apply to the problem. Once we put the problem in the middle, we could see what was missing. And what was missing was the model. We needed a laboratory cancer of what makes cancer cancer. We needed cancer. We needed a way to apply our tools. So we ended up starting with manufacturing. And the issue with manufacturing was, up until then, largely the research that had been done in 2D. The reason is you could see it. It was flat, it fit on microscopes, it had this ubiquitous infrastructure that we all used. The truth is we were all strangled by that infrastructure. We had come to rely on it so much that it became a constraint to the studies that we were going to do. And we needed to break that constraint, and we needed to go to 3D. To tell you the difference, all of us are soft, wet, and squishy, mostly water. We're pretty daggone close to the jellyfish. We're a lot closer to that jellyfish than that plastic dish, which is basically millions of times stiffer than the materials that we're studying. So we knew we needed to go into 3D. The problem is that 3D had also been tangled by compromise. You could either do it in a liquid, like a liquid drop, or by agitating things, where you could solve the transport problem, meaning you could feed the cells. Or you could do all sorts of things by putting them in gels and scaffolds. And then the problem is you had to deal with diffusion, and you had to deal with all sorts of suboptimal transport problems. So we asked an incredibly stupid question. If the liquids aren't optimal and the solids aren't optimal, is there a liquid-like solid? Every now and then a dumb question has a great answer. And the answer is there is. It's been around us for a long time. Hand sanitizer is a liquid-like solid. The ball pits that kids play in when they go into the little kids areas where they sit in the balls and they throw the colored balls at each other, that's a liquid-like solid. Those balls don't just fly all over unless kids throw them. You can sit in there. We needed a ball pit for tumors. So to create that material, notionally, it's as simple as taking hand sanitizer and growing cells in it. So what is hand sanitizer? Hand sanitizer are these little microgel particles that you see here on the right. They're swollen in ethanol and fragrance, okay? And they turn into basically something that we can use to sanitize our hands, to kill microbes. What we had to do was take hand sanitizer and make it a cell culture material. So we had to swell it in liquid media. We had to reduce charge. We had to add a whole bunch of things to it. We had to tune the size to mimic a capillary bed. It took us years to do that. But we did all of that here, and it's really a collaboration between chemistry, physics, mechanics, material science, all coming together to make this material. What you see here on the right are these microgel particles with individual cells as the colored objects that are sitting here. So if you can believe that Bath & Body Works can put these small particles in there, I think you have to believe as engineers, we can put cells and tumors in there. And that's really what we ended up doing. And then the concept for how this would work was relatively simple. A patient biopsy comes in, there's millions of cells in that biopsy. We turn that into lots of microtissues. We suspend the microtissues into microgels. We add back in the immune cells, all of the therapeutics, and then we use imaging to study the outcome. And the image that you see right here is a really special tumor from an extremely rare case that I'll talk about a little later that presented here at the University of Florida. About 3 years ago, we got a call. And the call was, there's this new emerging pathogen. It's a zoonotic virus, SARS-CoV-2. We don't have any animal models for it. We have a major issue. How are we going to do this? The CTSI here, again under Duane Mitchell and others, they put effort in to take and seed combinations of technologies. They took what we were doing with cancer, they moved it in with people that were studying infectious disease. We combined it with emerging pathogens. We worked with pharmacology and big pharma, and we put together a way to create a tool to look for therapies to SARS-CoV-2. And this is a picture of human lung, and the red dots are the first infection from SARS-CoV-2. So again, it's an evidence of where the university can force and bring together these transdisciplinary approaches on current problems quickly. The first one was basically captured here in March of 2020. We had samples running by basically early May, okay? On the left is a glioblastoma tumor, which I think you're gonna hear about in a little bit. And what it's doing is it's growing. So when I first had cancer, I wanted to know, how fast is it growing? How long do I have? And the answers were always like, everybody's different. You have to understand. We're all unique individuals. We don't have a good answer for that. As an engineer, it was frustrating. It's like, well, let's take 6 or 7 time points. We'll fit the data and we'll figure out an answer to this thing. Well, here we're able to do these things in the lab where we can watch this tumor growing over hours. The way this works, if we look over here on the right, is basically as that tumor is growing, it's pushing those microgel particles around. So now we've got a system that we can study the tumors, we can allow them to grow, and we can study their kinetics and their dynamics. The problem that I think a lot of you have probably thought about is, if it's in a solid, how do you feed it? In liquids, basically things are always in motion. So if you add something to it, it all mixes up. In solids, it all diffuses. So although we had everything working at this point, long duration culture was a struggle for us. So we invented Darcy plates. Darcy plates are extremely simple devices that do nothing but take a little negative pressure, draw the fluid through, and allow these tumors and tissues to basically grow in 3D culture. We built all of these here at the University of Florida. We also moved all of this out to Aurita when it graduated. But the issue that we had is that we had done all of our development for the single PI laboratory, the single investigator laboratory where everything was done by hand. Today, we've got these materials in big pharma, and big pharma wants to do these things at tens of thousands of wells per day, hundreds of thousands of wells per day. So we had to miniaturize our Darcy plate into a single well that you see right here. And this is really where having a company like Aurita was very useful, because this isn't an obvious PhD project. This is an engineering task. So now we've got a company that can do the engineering task, connected to the universities that can basically do creative design. We have that connected with big pharma that can define the need. We have that with our colleagues in medicine that are looking for therapies, hospitals that are providing tissues and patients, and in an ecosystem that's basically all coming around to try to find success. And this is what these new plates basically look like. So I just want to tell you a little vignette, and then I'll shut up so that I can listen to Duane talk, and I'll get off the stage as fast as possible. But this is how far things have come. A person presents here with a colorectal cancer, unknown. It has a POL mutation. It's an extremely rare mutation. In fact, it really shouldn't be possible in this particular tumor. We get the call. We've got a rare tumor. We've got a rare case. It's hardly studied. We don't really know how to treat it. Can we get it in the system? So, of course, the answer was yes. So when we look at this, basically after day 4, what we recognized is a really unique structure. And I'm just going to highlight it for you. I'll go back and talk about it. But if you look at these donut type shapes that are developing, as engineers, we recognize this immediately. This is a solution to growth without vasculature. This is a very, very, very fast growing tumor. We've only seen one other tumor that does things like this, and we'll show it to you next. So we recognize this as what we would describe as a very hot tumor. When we went and we did the staining, not that you're all biochemists or anything like that, but what happened is this tumor was full of immune cells, which meant this tumor should be a great candidate for immunotherapy. The thing is, the cancer type, microsatellite stable colorectal cancer, is not indicated for immunotherapy. And yet every indication we saw is that it should. So what we could do in the lab for basically no cost is run a synthetic immunotherapy trial. And that's exactly what we did. And what we found is that the POL1 mutation, the POL mutation overrides the microsatellite stable presentation, which says these tumors should be treated with immune checkpoint therapies. There's not enough people with this to ever run a clinical trial. There's not enough people that you could go out and do that. But in this personalized way, we're getting readouts on day 4. Present with a tumor, give us 2 weeks, we can start to say whether or not we think these things have therapeutic options. Does it mean we find a therapy for everybody? No. But does it mean we should try? Yes. So how are we ever going to validate this? So this again comes from Elias Sawyer, the group with Duane Mitchell and others. And what you're going to see on the left is a glioblastoma tumor. This is a brain cancer. What it's growing in is a brain mimic that is a liquid-like solid that we have coated with collagen-1 and proteoglycans to mimic brain. And this tumor is spreading out and invading. And now the challenge is that once we've brought that into the lab, bringing the therapies in to treat it and cure it. We just recently were awarded an FDA grant here at the University of Florida to run a bench and bedside clinical trial. It's 2 clinical trials. One's for the patient, and one is that we go in lockstep to try to validate these technologies so that one day we can run the clinical trial first. So then when I go in as a cancer patient, my first oncology meeting, gives me chills to talk about it, if not tears, my oncologist asked me what I wanted to do. I'm an engineer. If you came to me and asked me about a problem that you were having in engineering and you were a medical doctor, I would not ask you what you want to do. I would tell you things we can do. It seemed ludicrous to me, but I understand it now. I didn't have a lot of options in 2013. I am here because of immunotherapies. But wouldn't it be better to go to your doctor and have them say, we've looked at your tumor, we've tried 10,000 different therapies, and we think these 5 are our best options? I really believe 10 years from now, 20 years from now, that's going to be more likely the discussion you have than a discussion that says, what do you want to do? And I think the University of Florida is a type of place that these types of things can happen. It fosters those interactions. This is what graduated out of an engineering lab. Stuff. It's the stuff that Aurita can provide to other brilliant people to put together to work in 3D. It's the stuff that Big Pharma is already currently using. 1 year out, Aurita is profitable, working with Big Pharma, developing new technologies, and working with other places. The fields are enormous. The little devices aren't the field. They're things that you put together to try to help. The way that worked is an ambassador program. It's the way we all do research. We collaborate across universities, big pharma, medical schools and research hospitals, and disease-focused research centers. That's what universities are great at. Plugging in the companies, the people, the government, and everything else together, that's what helps this stuff go fast. So I really want to get off of the stage right now and let Duane come up and speak. And I really want to thank you all for your attention. It really has been a blast being here with UF and working with all of you. So thanks.
Greg, thank you so much. It's a tough act to follow, but I think my friend Duane Mitchell, he's another one of our most valuable players here at UF. So Duane, I'll turn it over to you. Tell us about iOncologi.
There we go. We got it. Great. Well, good afternoon. I want to thank the organizers, UF Innovate, and the organizers of this forum for the invitation to speak. And I always enjoy hearing Greg tell his story, and it is really a testament to the extraordinary environment we have here at the University of Florida. I'm going to tell another narrative about collaboration and bringing preclinical discovery into clinical treatments for patients with cancer, and about company iOncologi, that is a biotech spin out from the University of Florida focused on immuno-oncology applications for refractory cancers. Just by my background, I'm a professor in the Department of Neurosurgery here at the University of Florida, and a co-founder of iOncologi with my colleague, Dr. Catherine Flores, who's here in the audience as well. And our laboratory focuses on identifying new opportunities for immunotherapeutic treatment for refractory brain tumors, both predominantly primary brain tumors in adults and children, as well as metastatic brain cancers from lung cancer, breast cancer, and melanoma. So iOncologi is a biotech spin-out that is focused on developing novel immunotherapeutic approaches for refractory cancers. We have two really platform technologies that the company has licensed and has exclusive rights to commercial development. And the first is an actual RNA nanoparticle vaccine, which many people have become familiarized with due to the COVID pandemic and the success of the COVID mRNA vaccines. Our laboratory has developed a technology that uses mRNA isolated from tumor cells from a patient's own cancer to develop personalized mRNA-based nanoparticle vaccines. The second platform technology actually is a novel immunomodulatory stem cell therapeutic, which is an intravenously delivered stem cell therapy that actually overcomes resistance to immune checkpoint inhibitors, which are the most really effective class of immunotherapies that we have to date. I'll give a little bit of background on that. But despite the significant advances in treating cancers with immunotherapy and with immune checkpoint inhibitors, currently 2/3 of the patients who are actually treated with these drugs do not derive benefit due to either acquired resistance or inherent resistance. And the immunotherapeutic stem cell platform is focused on overcoming resistance to immune checkpoint inhibitors. Our group has extensive experience, and this is again a testament to the unique environment here at the University of Florida, with capacity to do preclinical discovery all the way through early phase and even phase 2 clinical trials with technologies that have been developed within the laboratories here at UF. Our group and myself over the last 20 years has worked in this field and has developed 5 different FDA-approved INDs for investigational new immunotherapeutics that have moved into clinical trials. And here at the University of Florida, we have spearheaded several first-in-human studies from technologies from our laboratory, including the platforms licensed by iOncologi. And so we think we're well poised to bring really an innovative class of biologic therapies into the field of immunotherapeutic treatment. So by way of background, many of you may have heard of immune checkpoint inhibitors, but really these are drugs that take the brakes off of the immune system and allow a person's own immune system to respond against the alterations that are present within their own cancer. And for cancers like metastatic melanoma, lung cancer, and now over 15 different indications that have received FDA approval for treatment with immune checkpoint inhibitors, this has really revolutionized the field and the treatment options that patients with advanced cancer have available to them. We can actually talk about curing metastatic disease using the immune system when just a decade ago, a little over a decade ago, this was still just a hope that we could leverage the power of the immune system to treat cancer. However, despite the remarkable success of this class of drugs, and is now in the US approaching a $20 billion a year market for immune checkpoint inhibitors and approaching 40 worldwide, still 2 out of 3 patients who are treated with immune checkpoint inhibitors actually don't derive clinical benefit. And so through some novel preclinical studies, Dr. Flores, who's a stem cell biologist by training and working in our group studying immune resistance, made a seminal discovery that we could overcome immune checkpoint inhibitor resistance using an intravenously delivered immunomodulatory stem cell therapeutic. And I'll show some of that data, but much of this has been published in really elegant manuscripts that are available in the literature. Additionally, Dr. Sawyer, a pediatric oncologist in our group working on mRNA nanoparticle vaccines, has developed a potent platform that really reprograms the immune system within the immunosuppressive microenvironment and leads to robust sensitization to immune therapy treatments. And so iOncologi has licensed both of these platforms for further commercial development, but due to the real capabilities and capacity here at UF, both of these platforms have reached a clinical stage of evaluation already, with FDA-approved INDs here at the University of Florida and the initiation of a phase 1/2 clinical trial for the RNA nanoparticle vaccine platform, and a protocol that is under development and already FDA IND approved for the stem cell immunotherapeutic platform as well. This is just a diagram of the RNA nanoparticle technology. What we actually do with this, see if this will, is to isolate RNA. You heard about the biopsy specimen that contains millions of tumor cells. We know that each patient's tumor is unique, but the mRNA from those tumors is essentially a blueprint of the antigens and the proteins that the immune system could potentially recognize. Through a novel formulation that encapsulates mRNA from tumor cells into a nanoparticle carrier, these RNA nanoparticle vaccines are delivered intravenously and have been shown to lead to a robust immune activation in all of the immune organs, as well as within the actual tumor site itself, to turn what is an immunosuppressive or cold microenvironment to what we would call an inflamed or immune-supporting hot microenvironment. Additionally, these nanoparticle vaccines activate the dendritic cells of the immune system, and these are the cells that tell your T cells what to attack. Through a series of preclinical studies that have been published in journals such as Nano Letters, ACS Nano, and others, Dr. Sawyer and colleagues have really shown that this is a robust platform for treatment in a number of preclinical model systems, and then has moved these studies into large animal studies and early phase clinical trials. This is just a snapshot of a handful of those models of head and neck cancer, melanoma, sarcoma, and metastatic lung cancers, where you're looking at tumor growth curves or survival curves of animals that are either untreated, treated with immune checkpoint inhibitors alone, or treated with the nanoparticle vaccines either alone or in combination with checkpoint inhibition. And in all of these models, which are largely resistant to standard treatment regimens or immune checkpoint therapies, you can see robust sensitization and prolonged survival using RNA nanoparticle vaccines. After small animal studies, due to the existence and a strong collaboration with our veterinary medicine college here at UF, Dr. Sawyer was able to advance these studies into canine studies, and these are pet dogs that have actually developed spontaneous brain tumors and are undergoing treatment at the veterinary medicine school. Through a regulatory approved protocol, these animals are typically just given comfort measures and euthanasia, but they were enrolled on a clinical study where they received an RNA-based vaccine made from a biopsy of their tumor cells, and this led to robust immune activation in this large animal model, and also prolonged survival compared to historical experiences with dogs with malignant brain tumors. This is currently also being evaluated in cats who have developed head and neck cancer and shown similar both immunologic and clinical responses. Due to the preclinical, both laboratory, preclinical data, and large animal studies. This was used in an IND package that was submitted to the FDA and has led to approval of a first-in-human clinical trial in patients with newly diagnosed gliomas, both pediatric and adult, and will be supported through the Pacific Pediatric Neuro-Oncology Consortium for a US-wide phase 2 clinical trial. The immunomodulatory stem cell platform, which is our second core technology, really came from a really stunning observation that the intravenous injection of a subpopulation of stem cells could reverse immune checkpoint inhibitor resistance across a now large number of cancers. And Dr. Flores, and through an elegant series of publications, has really dissected a unique mechanism by which stem cells of the bone marrow can be harvested to overcome immunotherapy resistance. I'll just show an exemplar. Again, this is of a malignant glioma that is completely resistant. If you treat these tumors with checkpoint inhibitors, you see no benefit in survival over untreated cohorts. But in combination with an intravenously delivered stem cell subset, you can more than double median overall survival and lead to long-term cures in this completely refractory tumor model. This data has also been replicated in other cancers. This is an example of a metastatic breast cancer, triple-negative breast cancer model, and non-small cell lung cancer model. And again, through a series of preclinical development and IND-enabling studies, this platform has recently received FDA approval to enter into a first-in-human clinical trial of neoadjuvant treatment in patients with recurrent malignant brain tumors. In this case, patients that are scheduled for surgery will receive combination stem cell and checkpoint inhibitor treatment, surgical resection, and continued treatment afterwards, allowing for the examination of intratumoral biomarkers that we have identified in preclinical studies as being indicative of clinical activity. We have under development an additional protocol looking at a similar approach in refractory solid tumors that have already failed checkpoint inhibitor treatment. The leadership team for iOncologi consists of the co-founders, myself and Dr. Flores, as well as a partner, Dr. Cash, who is an experienced biopharma developer with over 30 years of experience developing oncology drugs, several through FDA approval. And we have several core platforms of biologics within the portfolio. The IOI-7 are the autologous stem cell therapeutics, which are entering clinical investigation here at UF. IOI-8 is actually in preclinical development of allogeneic off-the-shelf stem cells that would be HLA matched to patients and ready for immediate treatment. IOI-10 is the line of RNA nanoparticle vaccines initially focused on personalized RNA isolated from patients' own tumor cells. And due to the advances already here at the university, the co-founders have received over $10 million in funding to support these early phase clinical studies. So we anticipate completion of phase 1 and phase 2 within 3 to 5 years of capitalization of the company, and then at that point, strategic partnership or acquisition for continued commercial development of this novel class of immune therapeutics. I'll talk a little bit about some of the competition and how we've positioned ourselves, we think, to fare well. This is a very crowded space in immuno-oncology, and a lot of combination therapies are being explored. One of the huge advantages we see to the stem cell technology is it's completely non-toxic in its delivery. And there's extensive experience with the delivery of hematopoietic stem cells to patients with hundreds of thousands of subjects having been treated. And therefore, this combination treatment actually is not anticipated to add any toxicity to the already known profile for immune checkpoint inhibitors. That has been a major limitation in many of the other combination approaches that have actually been limited by the additive toxicities brought together by combining checkpoint inhibitors or other drugs. The nanoparticle technology platform, many are familiar with, is extraordinarily versatile in being able to manufacture, synthesize, and distribute RNA vaccines. And it's also quite pliable in terms of adding other RNA constructs to enhance efficacy. And so again, we think this is a novel platform protected by a wide portfolio of patents that gives us freedom to operate in this space quite effectively. Market by the numbers, I won't spend a lot of time talking about the markets, but just to say that the immune checkpoint inhibitor market is projected to reach about $56 billion within the next couple of years. We believe that a single indication for either of these therapeutics is certainly over $1 billion US market, and a wide number of patients that this is potentially applicable for. Our initial evaluation for PD-1 refractory tumors is started in glioblastoma, with preclinical development for metastatic triple-negative breast cancer and lung. And we envision doing a series of signal-finding phase 1 studies to identify indications with the most robust clinical response for then continued evaluation. In terms of risks, cell therapies certainly have some risks around manufacturing, and fortunately in the stem cell arena, there is an extraordinary amount of experience with both collection, distribution, and scaling of hematopoietic stem cells, and so we think this is a huge advantage to leverage that existing experience across many institutions. Also, the pricing models for stem cell therapeutics are quite robust, and so we have an expectation of both the manufacturing costs and the expected reimbursable costs for this type of therapeutic to base it on the current markets. For RNA nanoparticles, there's obviously a lot of competition in terms of the RNA nanoparticle space. We have novel IP in how our RNA nanoparticles are formulated, and obviously have talked to several companies about potential partnership who have interest in this space as well. So in summary, the founders have really developed novel stem cell therapeutics and RNA nanoparticle vaccines that can treat refractory cancers that have reached a clinical stage of evaluation. We have quite a bit of experience in clinical development as well as biopharma development within the existing team, and I think a unique opportunity to bring a novel class of biologic therapies into the immuno-oncology field with broad applicability as platform technologies to refractory cancers. So thank you for your attention, and look forward to answering any questions.
All right, thanks a lot, Duane. Next up, we have Edgar Rodriguez, who is one of the great scientists at UF, but also has made a transition to be a great CEO. So Edgar, tell us about Lacerta.
Thanks to all of you for being here, and also to UF for the invitation. I've been around UF for a while. I was a graduate student here, left, came back, was College of Medicine faculty. And if you want to know about me, I was at UCF finishing my undergraduate degree when a professor from the University of Florida came down in 1998, so a while ago, to show this amazing result where he was able to use a virus to deliver a molecule that fixed vision in a rat. And so to me, that was just amazing. Like, I just couldn't believe that you could use a virus to deliver genetic material to fix, a rat that otherwise was going to be blind. And this was back in 1998. I'm originally from Puerto Rico, so you can imagine, I go to my Puerto Rican parents, I know exactly what I want to do. Please just sign a check every month. I'm going to go to the University of Florida, and I'm going to work on something called gene therapy. And so that was back in 1999, when AAV gene therapy was nothing but an episode on a Star Trek show, whatever was being shown at that point. And so since then, my trajectory has taken me through graduate school, working on ways in which we can design AAV viral vectors to deliver genetic medicine into the brain so that we can fix neurological diseases. And so out of that trajectory, eventually I made it here as a faculty member in the College of Medicine, and got an opportunity to take those technologies that we initially started working back in 2000. Took a while to get that, make it be a reality and make it be something that my parents could understand what the hell it is that I was working on, and actually now turn it into a company. Eventually moving from academia into the biotech, I really credit the ecosystem here in Gainesville and the University of Florida. I was provided with all the opportunities and support to be able to make that transition. It's something that I had not trained for, but was successful in doing. And this was back in 2018. Now in 2022, the environment is even better. So there's no better place for VCs to be active than in Gainesville, Florida, where all the support exists, not just the technologies, but the people who can bring those technologies and develop them and make them a reality. And so Lacerta Therapeutics provided that platform for me. I was the founding chief scientific officer, neuroscientist by training. and then eventually grew to become the chief executive officer of the company. So I discuss AAV gene therapy. What is AAV gene therapy? The use of this approach and this technology, in summary, has revolutionized really genetic medicine. And I think it's important to highlight these 3 products. So these products are 2 of them FDA approved and EMA approved. And then the last one here on the left by BioMarin was recently approved by the EMA. And what I'm trying to highlight here is how diverse and powerful is this technology, right? We can utilize this technology to fix eye diseases. We can use this technology to work on CNS conditions. And we can also use this for blood disorders. And all of it based out of the same technology. Oh, and by the way, this technology was invented right here at the University of Florida. And so it's really a revolution, but that revolution, just like many other revolutions, has a lot of complexities, and there's a lot of work that needs to be put into it and a lot of expertise. And thankfully here at the University of Florida, not only do we have the expertise in terms of the faculty that are working on these technologies, and a little bit later we're going to hear from Shannon Boye, who's one of the pioneers in this technology as well, but also the PhDs and postdocs that are being trained here that then help companies like Lacerta to be able to recruit and continue to mature some of the products that we've been working on. And so what are we doing at Lacerta? As I mentioned before, Lacerta is a spinoff out of the University of Florida. And what we did was we decided from the get-go that we wanted to work in 3 different specific areas of AAV technologies. One area would be focusing on how do we make this virus, this complex biologic, how do we grab our hands around it? How do we become engineers to actually be able to design the virus so that we can control its biological properties to the best of our ability, right? Predictability is a key aspect to any drug that you manufacture. The other aspect that we wanted to focus on was manufacturing. So not only how do we design it, but how do we manufacture this in a very effective way? And from the get-go, we knew that we needed to move into something that's called producer cell line platforms. And we wanted to make sure that whatever system we chose to work on and whatever platform we would choose to bring forth would be a platform that would allow us to bring the cost of manufacturing this product as low as possible. As I mentioned before, I come from Puerto Rico, so as I lead this company, one of my main focus and goals is to make sure that this technology is affordable and available to everyone who needs it. Lastly, we wanted to focus then in using those technologies, right, the engineering of the vector and the manufacturing platform, to be able to design drugs that can treat central nervous system related diseases. The company, this is just sort of highlighting, has been very successful. Not only is an ability to recruit pharmaceuticals such as Therapeutics, UCB Biopharma, which is a large European pharmaceutical company, as well as Eli Lilly, we've been able to establish solid partnerships with this company. And this really what's helped here is these partnerships validate the work that we are doing, right? But more importantly, it allows my guys to be exposed to large pharma, to work with large pharma, to train large pharma without necessarily having to leave Gainesville because they're looking for that type of exposure somewhere else. And so in many ways, it's an opportunity that Lacerta has really leveraged to be able to bring these names down here into Gainesville, down to Florida, so that they can get exposed to our expertise. And that obviously then leads, indirectly to more VC funding activity because there's validation of the technologies that we're actually developing. And so today, I wanted to take this opportunity to just highlight one disease that we're working on. And I'm mentioning one disease, and this is just, I guess, to get back to the fast break basketball. This disease is allowing us to do our fast break, right? Setting up our shooting guard, we push the ball to the side of the court, waiting for the shooter to come along, ball outside, ball inside, score. That's what this disease is allowing us to do while we set up our bigger offense, right? Lacerta's biggest offense is going to be attacking larger CNS indications where there's, these afflictions such as Alzheimer's disease, Parkinson's disease, chronic pain, things that a large portion of the population, has to have the challenge to face with. That's going to be our offense. And so here we're going to talk about our fast break. And that disease for us, our focus has been on Friedreich's ataxia. And this disease is an indication, what we call a well-suited indication for AAV gene therapy. And why is that? It's because it is what's called a monogenic disease, meaning we know exactly the gene and the mutation that leads to the disease. And so if we know what the gene is and we know what the mutation is, then AAV gene therapy is immediately the solution that you want to think about. And so in this case, this disease is caused by a defect in a gene that is called frataxin. And what that gene does is, going back to our basketball analogy, basketball players need a lot of energy. So what this protein does is it has an essential function in a little organelle that's called the mitochondria in your cell. And that mitochondria obviously is your energy storehouse and your energy warehouse and your energy producer, right? So what this, if you don't have the function of that gene, in this case frataxin, if you lose the ability to express that gene, then you lose your ability to properly energize certain organs and certain functions across your physiology. And so you can imagine, specific organs in your body that need a lot of energy, right, routinely, the heart and the CNS. And so patients that have this mutation where the frataxin gene is inactivated, it actually then causes issues that are related to cardiac hypertrophy as well as to degeneration of specific areas in the CNS. And I don't want to get into the weeds of exactly what the mechanisms in here are. I just simply want to just present to you that when we are thinking about treating Friedreich's ataxia, you have to think about treating the central nervous system as well as the heart of these patients. So the first thing that we wanted to address in this disease then was how can we target the cardiac component of these patients. So here I'm just showing you a quick picture of a heart that is hypertrophic. This is from a patient, an FA patient. And if you look at a normal heart, you can see that, tissue is nice and thick around that left ventricular wall. In this case, you have this degeneration that occurs in the tissue. And so our challenge when you're doing gene therapy, specifically with AAV, is can you deliver now a functional copy of that gene, right? As I said, frataxin is not working. So what we want to do is use AAV like a FedEx truck. And we're going to pack into that FedEx truck proper genetic copies of that frataxin gene. Then that FedEx truck is going to drive and is going to deliver that genetic payload to the heart, right? And so one thing that people started observing very quickly, and by people I mean other competitors, was that, yes, this appears to be a simple thing. Just put the package in the FedEx truck and tell it to go. But what people were seeing is when you do that with designs that don't take into account where the package is going, you start losing actually the therapeutic efficacy. And indeed, what happens is that it turns into a toxic insult. So what I'm showing you here is, the weight that is normally gained by a mouse over its lifetime. So this is its age of a mouse over weeks. And you can see that that mouse continues to gain weight over that period of time. This is just a normal mouse. If you have a mouse that has been engineered to display the cardiac anomaly and phenotype of Friedreich's ataxia, you can see that that mouse model begins to die already by about 10 weeks of age. These animals actually have a cardiac phenotype that causes them not to be able to pump blood. And so by 10 weeks of age, all of those mice invariably die. And what you can see here is that other competitors tried to use AAV gene therapy to deliver a cargo of frataxin into the heart of those mice. And you get this initial response, but eventually then you get a toxic insult. And so you never can get those animals to just behave like the normal animals that are highlighted here in blue. And this is a study that was done by Pfizer, published by Pfizer Pharmaceuticals, and then there are other groups who have faced similar problems. What we've done at Lacerta was actually think about the design on how that cargo is going to be delivered. And so what I'm showing you here is results from our specific engineered therapy, in which we can again see those animals here that are untreated invariably die by about 10 weeks of age. And what you are observing here in terms of the probability of survival is a dose response curve. So our product, as it is being dosed up, has now 100% rescue of those animals. And so we have a very broad therapeutic window that causes zero toxicity in these animals. And we can actually then measure expression of that frataxin protein in those hearts so that we can correlate the amount of expression that we have with the survival of these animals. And this is key for us, but that's mouse studies, right? We then needed to move these studies out into a larger animal system so that we can then really understand, in the context of a larger animal, will our doses still work effectively and safely? Not only that, but then can we actually detect whether, if we're expressing for 3 months or for 6 months, can we detect any toxicity that may be induced due to the treatment that we're trying to provide. And again, I'm showing you here results from large primate models in which we are infusing our drug intravenously at 2 different doses, demonstrating again that we can express our protein to about 80% at our highest dose, to about 80% of the wild type levels, which is more than sufficient of what we would need in a patient, as this demonstrated here in a graph, where if you analyze the patient population for FA, you can see that the protein levels of frataxin in the patients are about 25% of a wild type control, and the carriers, meaning the ones that don't develop disease but carry a version of the mutation, have about 65-70% of wild type levels. So as long as we're in that range, then we should be able to turn these patients into these carriers who are now not patients anymore. They don't have any clinical symptomatology. And so we think that we already have that dose, and more importantly, we know that our construct is working extremely well because we can analyze the hearts of those primates, we can analyze the livers of those primates, and we can show that whether the primate simply received a saline control or received our active drug, there is no difference in the health of those tissues. We've also looked at it via echo MRI, and we've had pediatric cardiologists do an analysis to make sure that there's no toxicity associated with this. And so quickly, again, just returning to the competition, when it comes to developing this drug as it's making its way to the clinic, you can see here that competitors, when they're analyzing their heart tissue, this is again heart tissue from primates, after they're delivering a similar, but sort of a similar product that is different in terms of the design, you can see here in this dark red color, these are immune cells that are attacking the heart. And what this is, every time you see this, this is a big no. So what this is showing is toxicity. In fact, one of these animals had to be put down early on because of the toxic insult from that product. You can actually measure that in the blood. You can look at an enzyme called troponin I, and if you see these spikes coming up, then this is indicative of a cardiac insult in these animals, again, demonstrating toxicity from that product. This is our product, boring, quiet, exactly how you want to see it. The other challenge that we had was, again, now targeting the CNS. Now, mind you, if you just look at this product, Lacerta right now has a product to be able to at least address the cardiomyopathy pathology in this disease, but that's not enough for us. We want this product to actually work also in the CNS manifestation of the disease. And so again, we go back to mouse models where we can deliver very effectively into the brain, express the protein in the areas of the brain that are needed so that we can have some actual functional recovery in the patients. And we can, again, take a very robust, aggressive model of the disease that is specific to the CNS, and we can demonstrate that in contrast to an untreated animal, which rapidly declines over a period of weeks in its ability to coordinate its motor function. You can compare this red line to a black line, and what you do here is you take these mice and you put it in a rotating rod, and you measure the ability of the mouse to balance itself in that rotating rod. You can see that the FA model lacks and progressively it diminishes its ability to maintain balance in that rotating rod, just like the disease, a progressive disease. And what you can see here is that if we actually treat those animals after their phenotype has now started, which is critical, right? Because this is around the time when patients are actually coming into the clinic. If we treat at that time, we completely now block the progression of that disease when compared to an untreated animal. And we have other effects that are very positive, such as maintaining body weight when compared to untreated animals. So overall, now what we have developed at Lacerta, with the help of neurosurgeons here at UF and other institutions across the country and the globe, is an MRI-guided precision delivery method so that we can actually deliver this drug directly into the areas or structures of the brain, as well as the spinal cord areas of these patients. And so we've already now completed a number of studies in the primate, that's what you're seeing here, demonstrating not only the safety of the approach, but the efficacy in terms of the distribution of the drug to the particular target tissues that we want in the brain. So we're very excited about our fast break. Lacerta should be in the clinic by the beginning of 2024. We're now working through the regulatory agencies, processes, and manufacturing of the product so that we can make it into the clinic next year. It is on the basis of this success that we are now in the process of a Series B financing opportunity. And so for those of you who have any interest, feel free to talk to me afterwards. I really briefly wanted to mention manufacturing. For diseases that are more prevalent, as I mentioned before, we need to come up with methods by which we can manufacture this drug, which is very complex, very complicated to manufacture. We need to simplify that so that we can make that cost come down, and so that we can then allow this to be able to, break out into indications like Alzheimer's and Parkinson's, where now you have to treat millions of people. For example, Friedreich's ataxia is about 20,000 patients worldwide. In the case of Alzheimer's and Parkinson's, obviously, we're talking 50s and millions of patients worldwide. So the degree of manufacturing, or the scale of manufacturing from here to here, is very high X-fold. So what we have done at Lacerta with technologies that were initially developed here at the University of Florida is what we decided to do is actually make use of insect cells. And so what we're doing actually is we're taking insect cells and we are genetically engineering these insect cells to contain all of the elements that you need in order to manufacture AAV. And we can do this in a very precise and rapid way, which makes it practical for drug developers. But more importantly, when we do this genetic engineering, these cells, they don't even know that they have all these elements in it. The system is completely silent, and we induce it very easily by simply exposing this cell to a wild-type virus, a virus that actually knows to infect SF9 cells, or a wild-type, an endogenous pathogen of these insect cells. And so by exposing this cell to the virus actually turns on the machinery. So now we can start making lots of AAV, and because of the way we've genetically engineered the cell, this process actually amplifies itself within each cell. And so really the key technology advancements that we've had is that we are able to go from the genetic engineering step all the way to a research cell bank and to a 10-liter bioreactor setup in about 3 months. And that is unheard of in terms of the speed by which we can accomplish this. And because of this, now Lacerta is in conversations with not only large CDMOs across the country, but also other drug developers that want to license this particular technology to bring it in-house so that they can manufacture their own drugs. Again, this is the key finding here for us, is the fact that we were able to engineer a way in which we can not only half the time to grow these cells, but we actually doubled the amount of cells. So this is a total improvement of 4x, which is very difficult to attain in the context of drug manufacturing of biologics in the Pacific. So I'll leave you guys with that. I try to be as fast as possible because this is a fast break, but I will be around. So if you need me for anything, I'd be more than happy to slow down and emphasize any area that you'd like me to go through. So thank you so much for your attention.
Next up is Myosin Therapeutics, and unfortunately, Dr. Courtney Miller had a medical emergency and can't make it here today. But in the spirit of sort of being the next man on the bench, I will go ahead and run through her slides and do the best job I can. And then if anyone's interested, we'll certainly put you in contact with Dr. Miller. So Myosin Therapeutics comes out of the Scripps Research Institute. And on the 2nd of April this year, I think many of you have heard that Scripps Research Institute in Jupiter, Florida came under the umbrella of the University of Florida. And there's 42 scientists down there, principal investigators. Some of the most advanced biomedical research in the world is going on at Scripps. And Dr. Miller is one of the experts in the field of molecular nanomotors. And there's very few people in the world that actually do this kind of research. I'll also point out her two co-founders, Dr. Griffin and Dr. Kameneka, have extensive experience in biopharma at Merck before they formed this company. So the development of the drugs inside Myosin have all been done with the efficiency of a biopharma company like Merck. There's been about $15 million of NIH funding supporting this technology. There's a broad patent portfolio around small molecule drugs that they've developed targeting these molecular nanomotors. I'll talk to you mainly here real quickly about MT125, which is the lead compound targeting glioblastoma, as Duane, so eloquently told us about, a major unmet medical need. When you think of molecular motors, we often think of myosin, which is the protein that allows us to flex our skeletal muscles. But it turns out that there are literally hundreds of non-muscle molecular motor targets that could be drugged. And Dr. Miller and her team at Scripps have discovered many of these. And the lead compound, MT125, is targeting two things responsible for the growth and the metastasis of brain cancer. So they do this. We have Dr. Tim Spicer here somewhere in the audience. Tim, thanks for coming up, who runs the high-throughput screening lab at Scripps, where about half the biopharma companies in the world come to Scripps to run millions of compounds through this high-throughput screening center, and it's also used by the investigators inside Scripps as well. And that's what Dr. Miller and her team have used to discover molecular motor targets and then screen millions of compounds against those targets to discover drugs. Now, the field of molecular nanomotors is fairly new. This is a fairly new field of biology. There have been some notable companies that formed around it. However, I'll point out Myocardia, that developed a drug targeting one of these molecular nanomotors for cardiac hypertrophy. It was acquired by Bristol Myers for $13 billion. Cytokinetics did a $450 million deal, and then two smaller companies, Motor Pharmacology and Kinomics. Now, the team, to the best of their knowledge, is that there are no other molecular motor-focused oncology companies, even though it's those molecular motors that are responsible for the metastasis when cancer cells move, and then when they grow uncontrollably. So if you could target those, could have an effect on the condition. So that's what MT125 does for glioblastoma. As Duane pointed out earlier, it's been very limited, essentially 3 buckets of research and been done for decades in this field around immunotherapies, chemotherapy agents, and in some cases, angiogenic inhibitors. But of all this research, really outcomes for patients are no different today than they were 40 years ago. So It's pretty, you hear a lot of exciting things on the news at night about advances in cancer therapy, but a cancer diagnosis of glioblastoma is essentially a death sentence to this day. Current standard of care really increases life by 3 to 4 months, and it's often not a very good quality of life. So a major unmet medical condition. And current standard of care I mentioned. So what does MT125 do is it targets 2 receptors. Let me just go up here. That are called NM2A and M2B that are responsible for what Courtney likes to call the go and grow of glioblastoma cells. An important feature of this compound is that it's brain penetrant. You've heard about the blood-brain barrier. Often drugs that are given cannot penetrate that barrier, so even though they might be effective, it can be very difficult to deliver them. This drug appears to be highly brain penetrant and also can be delivered IV and perhaps even subcutaneously. So it wouldn't have to be given directly into the cerebral spinal fluid or in an open brain procedure. This is the go and grow between the 2 receptors that are targeted by this drug. So both of these receptors are inhibited. And what led to the idea of targeting the receptors was this kind of study where if you take a mouse and knock out those receptors, they tend to respond pretty well to glioblastoma and have a high survival. So that led the scientific team at Scripps to think about, why don't we develop a small molecule targeting those things, essentially blocking those receptors and in effect knocking them out in vivo. This graph here shows you that the compound is penetrant into the cell, and in vitro experiments seem to show that it's not toxic. Again, there's some data showing that it's able to block the grow, where the cells divide uncontrollably, as well as the go, where the cells are migrant and metastasize. And so these are some of the preclinical data that show what does MT125 do for survival. And here you can see nearly a doubling of survival when the drug is used alone against untreated controls. And then they began to think, well, could we get even better data if we combine that with current standard of care? And current standard of care is radiation. And here you see, in combination with radiation, even greater survival of these animals. And then for me, this is probably the most exciting thing that Dr. Miller's developed is in combination with a commonly known and commonly prescribed FDA-approved drug that is used in cancer, how did the results look? And in this case, over a 3-fold improvement in survivability. So again, these are preclinical studies. If this translates into humans, again, this would be a major breakthrough for patients. Some of the science team I already told you who founded the company, but they have collaborators they work closely with. I'll mention Steve Rosenfeld at Mayo Clinic. When I first became involved with Scripps and saw what Courtney and her team were doing down there, I saw that Steve Rosenfeld was involved, a guy I've known for a long time. He's worked in cancer therapy for over 20 years, and said, Steve, what do you think of what's the MT125 and what's going on? He said, I independently evaluated in my own lab, and the results were so profound that I repeated the experiments just to make sure they were real. So he's very excited about the potential for this drug. There are some other compounds that the company has licensed and are developing, but the lead compound, again, is in glioblastoma. However, there is some in vitro evidence that MT125 can be effective in some other cancers as well. It's an experienced team. They put together very detailed plans of what it's going to take to get this drug and how much money it's going to take to get it into humans. And right now they're developing a plan. It'll take about $20 million to bring this into phase 1. And in light of the sort of current market conditions, doing a smaller round to do IND enabling studies to get this ready to go in the clinic, probably around a $2 million round of financing. So that's the best Courtney Miller imitation I can do. And thank you very much. And thanks. And now I'll tee up our next speaker, Matt Donovan from Agriculture Intelligence.
Just click it, right? Well, good afternoon, everybody. My name is Matt Donovan. I'm the CEO and co-founder of Agriculture Intelligence and the Chief Product Officer for AgriView, a deep learning agriculture platform that was licensed right out of the University of Florida. I actually want to talk a little bit about the building we're in, the Stephen C. O'Connell Center. It's named after a past former head of the Supreme Court Justice, as well as the president of the University of Florida, who led this university from some turbulent times in the 60s and 70s, and I believe sowed the seeds for the better future that we see today. The O'Connell Center is also a place for joy, where entertainers from all across the world come, and of course we heard our hometown hero, Tom Petty, a little bit before for some intro music. This building, this place is a place of athletic performance, where national championships are won or sown. Go Gators. And most importantly, this place right here is a place that confirms scholarship and academic achievement by some of the brightest minds that Florida and indeed the world has to offer. So all these memories, all these achievements, all these accolades nestled right into 4 acres of North Florida goodness. Makes me want to say go Gators again. But 4 acres. This building is an enterprise. Just think about this small event that we're having here today and all the effort that was taken to put this together. Think about the timing to put on a concert here. Think about the planning, the logistics to get as many rowdy reptiles in this place on one of those SEC showdown basketball nights. It's an enterprise. It's an enterprise of 4 acres. And what I want to talk about is, what if that 4 acres was 10 times bigger? What if this place was 10 times bigger, or 100 times bigger, or 500 times bigger? The problem of scale is something that our platform is really here to address. So as small as we might feel in this room, just think if it were 100 times bigger. Anyway, here's a quick test. If this dot's a tree, how many? Pretty easy. How about now? And how about now? And how about now? You can barely see the dots. So the idea that even just something as simple as counting, when it meets scale, becomes very complex, very complicated. And in the field of agriculture, it has almost been incalculable. Just to give you some perspective, there's the O'Connell Center, situated in a relatively normal grove in a pretty mediocre citrus grove in Southwest Florida. So that's really what I want to talk to you about today, is how to tackle the problems of scale in a very specific agriculture area known as perennial tree crop growing. Now, most of you know corn and wheat and these kinds of things. They get lots of the oxygen, but breakthroughs have to happen to provide security for the food chain across all sectors of agricultural development. And here's a fact. When it comes to perennial crop growing, just the inventory, just the counting, can be off by as much as 25%. And in a marketplace, the global agricultural market is a $2.4 trillion marketplace. There still exists fundamental business problems to solve. The culprits are some flawed tech and processes. In the agriculture market, when you're trying to complete an inventory, you get a couple guys in a truck, you give them some clickers, and you send them out on a hot, humid day in the middle of Southwest Florida and say, make sure you get that count right. It's a slow process, it's expensive, and most of all, it's inaccurate. That inaccuracy has deep impacts. Garbage in, garbage out. Good information in, good decisions out. Bad information in, well, you know the rest. That's part of what the perennial tree crop market faces and has faced for decades. Real impact is that something as needed as an accurate inventory is the gateway to precision agriculture. Having correct information, as fundamental as that seems, is what will enable breakthroughs for advancements in nutrient tracking. Things like in-field work, and the ability to reduce chemical use all comes from the ability to accurately track what was there and what is to come. I'm happy to report that the AgriView platform solves those problems. AgriView uses multiple data sources, such as drone data or satellite data someday when the resolution's good enough, as well as ground data, as well as in-ground sources. And it uses a deep learning platform with computer vision, machine learning, and artificial intelligence to render real, accurate, proven results. So for a citrus grower, for example, we can tell them the tree count with precision of 98% accuracy. We could tell them the exact lat lawn, which is almost like a website address that I'll talk about in a minute, as well as the gap count, where there's missing trees. These are opportunities for that grower or for the industry to improve. Per plant health metrics, such as the canopy size, the density of the leaves for every single tree in the grove, no matter the size. As well as using ground sensors, which we've recently taken a license on, we can count and measure to the centimeter every single fruit in the grove to provide a true digital yield tabulation. It's not even an estimate for a crop grower. In addition, we have advancements in aerial-based leaf nutrient analysis that are on par or better than the classic sample tissue-based lab run samples, all in an easy-to-use web interface. Oops. So if any of you were wondering what was the answer, 236,809 trees, and about 80,000 gaps. So with AgriView at the center, we can immediately start serving growers with that accurate information. We can give them supply chain visibility as well as starting to develop true in situ yield metrics. We can help them understand harvest planning better. They can make better choices for inputs, which reduces the chemical footprint inside the growing area. And for their business needs, helps them understand the true yields that they have as they march towards harvest. On the financial side of it, we can immediately tell growers with accuracy, something that insurers in the agriculture market have missed for decades, but desperately want. The accuracy we can provide hits on the three pillars of any insurance model: accurate rates, precision underwriting, and fast claim assessment and settlement. In the emerging carbon markets as well, AgriView already provides a confirmed replant map and the temporal analysis that can track a tree's growth from not being there to being new to a fully mature tree, and including, as we move into the future, the potential carbon outputs of those trees as an improvement. For the grower, I'll simplify it. We're saving them money, we're giving them back time, we're saving their time, and we're showing them a path for better revenue growth. Now, growers are in a tough spot, so budgets are a little difficult. And while we're happy to support a modest number of growers here in Florida, we focused most of our initial efforts on the insurance market, where we knew we would have a high value proposition. I'm happy to report that we have a multi-year agreement with the #2 ranked U.S. crop insurer in the United States, NAU Country. There's only 8 of them in the United States, but NAU Country as a crop insurer represents the archetype of the crop insurers or crop mutuals that you'll find all across the world. I was super excited when they picked us over the 9 other funded competitors in a global search, but I'm happy to report that they picked us because of the proven and accurate and consistent results. And we did all this with under $1 million of funding. And our top 3 competitors in the field combined had over $150 million. And that's not something special I did, but that really is a testament to Dr. Yannis Ampetzidis, a professor here at the University of Florida and the inventor of the AgriView platform. We used every single dollar we could, and we delivered with accuracy that this company had never seen. The procurement officer told me that our accuracy beat the others hands down, and it wasn't even close. That's what UF is about. I'll be with you in a second. I'm trying to get the clicker to work. There you go. Yep, got it. This strategy actually is really important for us because it provides us with direct revenue. But for every single insured acre that we analyze, it gives us a sales opportunity with growers and other customer segments. The marketplace itself is very large, but just focusing on citrus in just 4 countries, there's a substantial market opportunity. By adding 4 other crops, just 5 crops in 2 more countries, the market opportunity doubles. By adding some advanced features to those 5 crops and focusing on just expansion in 3 other countries, we double the opportunity again. And this is just the 5 perennial crops that we've identified in 9 countries. There still remains over 30 perennial tree crops what we could serve. And the top 15 perennial crops that we see a gigantic market opportunity. Interestingly and importantly, we are in an active acquisition space with over 250 acquisitions occurring since 2020. We think we're poised to growth, but we have to know how to get there. So we want to focus on citrus and a full product we have, inventory, health, fruit count, and nutrient analysis in 4 countries, US, Mexico, Brazil, and Spain, with Brazil and Spain being our beachheads in South America and European markets. In 2024, we want to expand to those 4 other crop profiles just to do the counting. Nothing advanced, just counting trees. In almonds, avocados, olives, and grapes. And then we add Italy and Greece for obvious reasons. In 2025, we want to expand to providing those additional advanced features that from now until then, we'll be able to develop a precision level for nutrient levels and how to reduce chemical use in the fields for these top 5 crop profiles that we have. And then just adding in 3 of the most popular wine growing areas, France, Chile, Argentina, and of course the US. The most likely path for exit in the ag tech market is an acquisition. IPOs do happen, but they're rare. And we believe that we want to build a strong company with a great product, and we've got a great opportunity to make good revenue, but we think by 2026, we'll be in a good position to be acquired. Here's some income projections, which I'm going to zip through. Just know that this is a very modest single-digit market penetration for just those 5 crops. We have a small but experienced team. I myself have a deep background in the insurance industry, as well as serving as a CEO in a prior life, growing a SaaS software platform, very similar dynamics required to grow AgriView. But we're really led by the vision of Dr. Yannis Ampetzidis, a professor at the University of Florida and inventor of AgriView. His vision for what needs to be done regarding remote sensing and high throughput phenotyping is really something that makes him the leading global voice in the use of AI really in the world. We need to grow the company with some key team members, mostly in the sales, marketing, and people who understand the kind of international markets that we want to connect with. And we do have some advisors. This guy over here, Jackson, has been the voice of firm reason on occasion, but Dr. John Boye, who's also here, as well as Guy Latour, they're the kind of ecosystem that we have at the University of Florida that when a young startup is getting going, they need. And we've been lucky to get their advice when we needed it. We are raising money. We see this as a phased approach. We're first round of money that we're seeking in phase 1 is $2 million, and that's to build out some data acquisition strategies, improve our user experience so we can have a product-led growth strategy, which is critical for software revenue expansion, as well as those additional inventory crops. And then as we move along, we can either accelerate the process into other geographic markets, pick other crop profiles, and then do the R&D work to bring those products online. If any of you'd like the presentation itself, feel free to scan that QR code. My contact information there as well. I'll be available after here and at the event at the Hub. Thank you so much for the time. Thank you, everybody, for putting this event on. I know it's a lot of hard work. Thank you very much.
All right. Next, it's my pleasure to introduce Ben Patz from DeepWork Capital. And he's not the slam dunk artist. That comes later. I know Ben's tall. But anyway, come on up, Ben, and tell us about State of Florida Venture Capital.
Okay, thank you, Jackson. First thing to check, make sure I can advance the slides. So, again, my name is Ben Patz. I am a managing partner and a co-founder of DeepWork Capital. We're an early stage venture capital firm. We're super proud and pleased to be a sponsor here. Now, when Jackson asked me to come speak, he said, the reason that I want you to speak is because you're 6'6" and you're the tallest VC I know in the state. By the way, I expect you to dunk the ball. And I said, Jackson, that ship has sailed. It was a long time since I've been able to dunk a basketball. He said, okay, well, at least you're an engineer, and I trust engineers. You're going to be able to present some data to the team because this is the brightest audience in the world. And I said, okay, I'll be able to present some data. And then he failed to tell me that I needed to follow Max and Greg and Duane and everybody else here who are the absolute leaders in their field at what they do. So I will do the best that I can to provide some information on the state of venture in the state of Florida, how we see things, and how excited we are about that. So to start with, though, let me just tell you a little bit about us. Again, DeepWork Capital is an early stage venture capital firm. We were founded in 2015. Myself, my other partners, Mitchell Lasky is here, and Kathy Chu. And I always think great companies, and I consider DeepWork Capital a great company, come from a really great idea, but something that is brought personal. And so I'll tell you the personal aspect of this for me. So my kids all went to school in Florida, and they're all engineers and scientists. And my nieces and nephews are all engineers, and they're scientists. And I remember that when my niece went to go work for her tiny little company that she works for, and she said, I asked, well, why aren't you working in Florida? And she said, because nothing cool is done in the state of Florida. And I took that as a personal affront and a personal challenge to help build some really innovative companies here, and got actively involved in venture. And I started that with angel investing, and then being the engineer, said, this is crazy. I can see how you get money in here, but how do you help these companies really get to the next level? And that's where I met my partners, and we built a firm that is here to provide funding for companies advancing technology. We've done a pretty good job at it. Our first fund more than returned all the money back to its investors, and we have a dozen other investments still to exit. In addition to that, we've been awarded, as Mike DiNapoli said, the management of the state's venture program called the Florida Opportunity Fund. And so we have capital to deploy there. The bottom line is we have a significant amount of capital that needs to be deployed fairly quickly in super exciting companies, which I'm assuming now everybody's going to come talk to me right after I get off the stage here. So, oh, by the way, that little company my niece works for is Apple. But this is the current state of venture across the country. And if you look at that, it looks particularly depressing in the sense that over the last 3 quarters, there's been almost a 50% decline in the amount of venture investing that's going on in the United States. And there's a number of reasons for that. The one that's canonically given to individuals is, look, the rising interest rate environments makes investing in these early stage companies problematic, and therefore I'm better off doing something else with my money, and it's a drying up of that. But the reality is it was something a little different. And it's if you zoom back out on the data, what you see is that over years, venture investing is an up and to the right proposition. And last year just happened to be the single biggest year of all time in terms of venture investing in the United States. And if we look at this year, the disaster that we're having, we're still on pace to outperform 2020. And frankly, we're still on pace to perform. And that's something I learned in my engineering curriculum, which was things regress to the mean. This is just the way things work. And so if you have an outperforming year, it is going to go down, but whatever the mean is going to continue. And the mean here is a growing investment opportunity. Florida has behaved somewhat similar to this. It's been a slowdown in venture activity in Florida, but to a lesser extent. And the lesser extent is because Florida never saw the run-up of venture investing that you saw in California, in New York, in Massachusetts. One of the statistics that Michael alluded to earlier, it just depends how you slice the data, is that Florida represents, in terms of share of capital, under 3% of the share of venture capital. Now, I don't know about you, but simple math says that Florida should be north of 6% if you base it on population and north of 7% if you base it on GDP, where there's a whole bunch more capital that needs to get deployed in the state of Florida. And one of the things that was most interesting and exciting to me about what Mike brought up is of the 1,000 people moving to the state of Florida, there is a phenomenal wealth migration to the state. In fact, it is the single greatest migration of wealth and since the history of the beginning of this country, and it's migrating into the state of Florida at the exclusion of other places in the United States. So what you're going to see is a significant portion of that wealth being deployed into venture in the state of Florida. But nevertheless, what we find ourselves with is a scenario where, as Mike said, about 75%, 73% of all venture is done in 3 states. It's done in California, it's done in New York, and it's done in Massachusetts. And we need to change that. But why do we need to change that? You know, I mean, it's nice that we're small and we need to get bigger, but why? And so, again, being an engineer, one of the things we did in our company was analyze the data. And what's the data show? And so for this particular analysis, which we did last year, we sliced the market up into kind of 2 segments. We picked the top 5 states because that actually represented 80%. And it's just this nice little 80-20 thing we wanted to play with. Top 5 states, and we took everybody else in the bottom 20%. We said, let's look at the deals that actually happened there, but let's look at it from an investor point of view. What type of returns did they get? And so in looking at those deals, we found, we analyzed in terms of the exits, and big exits, $100 million exits. And the top five markets had about the same number of exits as everybody else. And in fact, if you look at unicorns, the billion-dollar companies, those were dominated by the California and New York and Massachusetts. And so you would just, on top of your head, say, why the heck am I investing anywhere except those markets? Because, my gosh, that's just where all the success is. And they analyze it just one more level, because the reality is you don't, as an investor, get a return based on what the exit was. It's based on the ratio of how much money you put in to how much money came out. And when you look at that, you find out that, well, wait a minute, the efficiency of capital at the median is about 2.2x, meaning that on average, these investments return 2.2x of the amount of money that went in if you were in California. But if you were in Florida, they return 4.96x. This state is one of the most capital efficient markets in the country. It's a good investment to make this. So not only do you see a market opportunity where we're an underserved market, but in fact, it's a great investment opportunity because we'll be able to return capital to the investors at a higher rate than they can do in other markets. This is why we built DeepWork Capital. Yes, it's to serve a need. Yes, it's to get my nieces and nephews and kids back to this state because we do cool stuff here. And you just heard 6 incredible companies do the most amazing things in the universe. But it's also, since we're running a capital firm, to make money for our investors and to do it better than anybody else does. And this is a sad part about the investments in Florida. These are the biggest successes in terms of IPOs in the state over the past 5 years. You might recognize some of the companies. I think Mike even referenced one of them, Luminar, on the top. It's an Orlando-based company. Okay? The total number of real investors is on the right there. The number of those investors that actually were in the state of Florida is on the left. It's less than 6%. So although this is a great place to invest, and we are dramatically more capital efficient than anybody else, guess where that money is going? Outside the state. Got to turn that around. And that's one of the fundamental objectives, again, of DeepWork Capital, is to attract capital in the state. Now, one of the other things that Mike didn't say really clearly with respect to the Florida Opportunity Fund, the $100 million of capital that's got to get deployed here extremely quickly. There is one overarching requirement of that capital. It must be deployed in a Florida-based company. I don't have the ability to deploy that in acute technology from Atlanta or from Chicago or from anywhere else. It's going here. And so I think this is going to help the growth in Florida. These are some statistics that I just think are particularly useful, and it's kind of why also we believe in this particular market and state. But it's some things you don't necessarily know or realize. We're the #2 medical device manufacturer in the country, #4 on high-tech employment. We have billions of dollars of research poured into the greatest universities around. And although I'm not a UF grad, I did go to RPI for undergraduate, as a couple of other folks here, and I'm phenomenally proud of that education. I did my graduate work at the University of Central Florida, and I'm phenomenally proud of that education, because in this entire state, we have incredible research at UF and at UCF and at USF and all throughout the state. We also have great other programs to advance this. And if you're not familiar with any of these groups, you should become familiar, whether it's the Florida Venture Forum, or whether it's the Tampa Bay Wave, or the other incubators and accelerators to advance these companies. You need to become familiar with those groups. So I'm going to kind of close on 2 topics here, and the first of which is going to be, so what do you invest in? And again, if you haven't heard it 6 times already, I'm an engineer. There's going to be an analysis. We're going to analyze the data. And what does the data say? The data says, relative to the other markets, Florida underinvests in healthcare. And by healthcare here, what we did was we lumped all of the healthcare services. So that includes biopharma, that includes medical devices, that includes health information technology. But we underinvest in there. Problem, because of regression to the mean, we should be at least up to that bar, which means opportunity. So that's where we're investing. And 50% of our capital in our last fund went into that space. And a number of investments of UF companies were made from our fund in that space. Also, it's a tech focused. So the deeper sciences, the harder sciences, are something that's grossly underinvested in the state of Florida, whether it's Matt Donovan's Ag Intelligence or Max's company that's building the next generation displays. These are companies that need to be invested in Florida. Last thing, Jackson promised I could include a rocket, because my career started off as a rocket scientist. I launched rockets for a living. So this is as close as I could get to launching a rocket. But there is some stats that I think everybody here, I hope, takes to heart about the state of Florida. And that is, we are the 3rd largest, home to the 3rd largest technology. We're actually the 3rd largest state in the country. We have hundreds of thousands, or thousands, tens of thousands of high-tech jobs in here. And look, this is the state that's adding the most technology companies. We need to be the funders of that, and that's what we're here to do. So thank you for letting me talk, and hopefully I did okay after the geniuses that presented before me.
Hi, everyone. I'm Stephanie Bales. I am the president and CEO of the Cade Museum for Creativity and Invention. And you might wonder why the Cade Museum is here. I'm only going to take a minute to tell you. First of all, I'm here to introduce Jackson and the one-on-one with the Atsena. But I'm also here to tell you a little bit about the Cade Museum. We are named after Dr. Robert Cade, who was the lead inventor of Gatorade. For those of you unfamiliar with the Cade, we are a mission with a museum. Our mission is to evangelize and support a culture of innovation and inventivity throughout Gainesville, the state, and we hope one day the world. At the Cade, we do this by first identifying inventor stories, their inventions, the concepts behind the inventions, and we do this through our Cade Prize for Innovation. It's now in its 14th year. There actually have been a couple of presenters today that have been finalists in the Cade Prize. And the Cade Prize for Innovation is important for the Cade because we take these concepts and we translate these concepts into cutting edge education that we then share with our community and throughout the state. We integrate these ideas into our STEM programs, our experiences that we offer in the Cade Museum and field trips. And if you've not had a chance to visit the Cade, I hope you would use the tickets that are in your bag to do so. I'd like to say that the Cade inspires the children of today to be the creatives and inventors and problem solvers of tomorrow. Who knows, we just might inspire your children to be like all of the individuals that we've heard with these cutting edge ideas that we've been presented to today. I don't know about you, but I have a lot of hope for our future just based upon the revolutionary technologies and ideas that we've heard. So now I'd like to introduce Dr. Shannon Boye, founder and director of Atsena Therapeutics. Atsena is a clinical stage gene therapy company focused on bringing the life-changing power of genetic medicine to reverse or prevent congenital blindness. Their proprietary assets are powered by adenovirus technologies, tailored to overcome the hurdles presented by inherited retinal disease. Dr. Boye received her BS in marine biology with a minor in chemistry and her PhD in neuroscience from University of Florida. She has been regularly recognized for her exemplary work in academia and is now the Associate Chief of the Division of Cellular and Molecular Therapeutics in the Department of Pediatrics, as well as the founder of Atsena. And Dr. Jackson Streeter, who you all know, but I'm actually a huge fan. He's an inventor of more than 20 patented technologies, the founder, as you know, of multiple startup companies, including Banyan Bio and CEO of Banyan Biomarkers, and now is the director of the ventures here at UF Innovate. And my favorite fun fact, the first naval flight surgeon selected as a Top Gun staff instructor in the Navy. So here's to you, Jackson. Thank you.
Thanks, Stephanie. So I encourage anybody who hasn't been to the Cade Museum, it's a fantastic place. Definitely should check it out. So rather than doing another PowerPoint, I figured we'd have another one of our outstanding scientists. I'm a huge fan of Shannon Boye, and just kind of get some perspectives from her, what it's been like to start a company out of UF. And Shannon, why don't we begin, like, where are you from and how'd you end up here in Gainesville?
Is this on? Yes. I'm from upstate New York, a suburb of Rochester. So I very quickly got tired of the gray snow awfulness of upstate New York. So I made my way to Florida as a graduate student. I got my PhD in neuroscience here in 2006. And actually my thesis project focused on developing a gene therapy for an indication that we've now brought into the clinic under the umbrella of Atsena Therapeutics. So it's been amazing to see that project go all the way through. But yeah, so I'm a migrant from New York.
Yeah, well, glad to have you. So, I mean, you were a bench scientist for a while, working, thinking about getting grants and, that's a whole path in itself. But when did you, and how did you start thinking about, maybe starting a company? I mean, when did all those ideas of entrepreneurship, come forward for you?
Yeah, so I work with my husband / co-founder of Atsena, and we started working together back in 2004. And we developed a number of technologies, both, to cure specific inherited retinal diseases, as well as optimizing the AAV vector itself. And we were naive at the time, and we thought the best way to get those technologies into patients that needed them was to out-license them to existing companies that already had the capital and the infrastructure to bring them across the line. But what quickly emerged over maybe 5 years' time was we were seeing these technologies not making their way into the clinic. And I think there were a number of reasons for that. We out-licensed to big pharma, to small companies, to medium companies. And when we partnered with big pharma, we would see everything get siloed into these different functions that didn't speak to each other. And things that should have taken a year and a half, were now taking 5 years. So that was incredibly frustrating. And then when we would partner with the smaller companies, we would see business decisions overriding sound scientific decisions. So that was equally as frustrating. So while the whole process was really beneficial, we were able to network with kind of everyone in the space on an industry level. We were frustrated, and we decided finally that maybe we should form our own company so that we could have a little bit more control over the direction that the projects took. Understanding full well, I'm not ever going to be in full control. We have a board and investors, but at least we could push things forward with strong scientific backing.
I mean, you kind of bring up a good point of something that Jim and his team deal with all the time with the OTL offices. Promising technology, should they go directly to pharma or into a startup?
Email, I was just irritated, and I'm like, why is this company taking this approach? Why is that company taking this approach? And he just wrote back, let's talk. So we got on the phone, and he said, have you thought about forming your own company? I said, yes, but I hadn't kind of said it out loud. And he said, well, let's talk about your IP. And so I talked with some of the folks in the OTL office, and we generated the list of IP that we had, and I showed it to Ben, and his eyes kind of widened, and he's like, you should really do this. So then I talked with Jim O'Connell, and the combination of Jim and Ben, really there was a mentoring process through how to do this. And so this was pre-pandemic, and one of the first in-person pitch I gave was in Cambridge at Atlas. And Jim came with me and got me caffeinated at some weird hole-in-the-wall coffee shop. He's like, let's get coffee before this pitch. I'm like, all right, I'll see you at Starbucks. He's like, no, it's this random address 10 miles away. But he got me a cortado, and then we went to Atlas, and they wound up passing on the technology. It was their loss, but we had a good time. But both Jim and Ben were there with me, so that was great having that moral support. And then Jackson, you came with me to Hatteras Venture Partners in North Carolina, and I think I felt the energy in the room that day. It was really good. I knew we had it, and I remember giving you the big high five when we were in the conference room afterwards. And then from there, it was just a number of pitches and the seed round and so on and so forth.
I mean, it's a credit to you to get really great groups, even getting a meeting with Atlas and Hatteras and those groups. And you've had some investors we'll talk a little bit more about. But I mean, you were very coachable, which made it real easy. And so it was really a pleasure to work with you on that. And so who did you end up ultimately getting funded by? And, can you walk us through that sort of Series A round with Atsena, right?
So our Series 1, we were invested in by Hatteras, by the RD Fund, which is a part of the Foundation Fighting Blindness, the University of Florida, of course, and Osage University Partners, and that was about $8.3 million. And that was right before the pandemic hit. So when it got time for Series A, it was completely virtual. I know some people prefer to pitch in person and get to know people better, but for me, it was so much easier because we had so many meetings and I wasn't on a plane, 24/7. So we did Series A in the summer/fall of 2020, and by December of 2020, we had raised $55 million. So that totaled 63 and change, and our Series A investors were Sofinova, Abingworth, and Lightstone, in addition to the seed investors, which came in
some of the
again.
greatest groups. I mean, it's great to do something for investors, but more importantly is you're doing something for people and families and patients. And could you just tell us a little bit about the technology and
Sure.
what it's going to do?
Sure. So we are an ocular gene therapy company. Edgar has already done a great job of describing what AAV is. It's the vector or the FedEx truck that delivers the gene of interest to the patient. In our case, we're focused on congenital forms of blindness, so blindness that affects, kids from birth. As I mentioned, I worked on one form of that kind of blindness as a graduate student. It's called Leber congenital amaurosis, type 1 specifically. And typically babies are diagnosed within the first few months of life because they're not fixating or on mom or dad, or they have a roving eye movement, which we call nystagmus. So I worked for many years to develop a gene therapy and evaluate its success in animal models of the disease and its safety in higher order species. And ultimately, we wound up partnering with Sanofi. That was the big pharma experience I had. Later on, Sanofi as a whole decided to divest itself away from ocular gene therapy because they had 2 other programs that were failing miserably, to be blunt. But we wound up benefiting from that hugely because we were able to bring the LCA1 program back. It was kind of a homecoming, so now it's with Atsena.
Yeah, it was great for us to participate in the round, and for myself looking at it in diligence, having Ben Yerxa and the Foundation for Fighting Blindness, which sees all the technologies, and they were really behind it, so it was a real kind of no-brainer for us to jump in. So through this process, what was sort of the most unexpected thing that happened to you, as a scientist turning into a business person? What did you learn?
Well, I did learn that in order to fly first class, you have to completely transition over industry, like Edgar, if he's still listening to me somewhere. I passed him on my way to coach the other day, and he was in first class, but I'm still a professor, so I sit and coach. Most unexpected. I thought about this a lot and how to answer it and how to do it in a way that I wouldn't inadvertently offend someone, so I'm going to do my best. I think what's been the most unexpected for me is the tightrope that you have to walk between educating the investors or the VCs that you're pitching to and not kind of insulting their intelligence. I feel like I spend a lot of time explaining why the snake oil that they were sold last week isn't the right way to go. And it's not to insult their intelligence. It's just you only have 45 minutes or something with these guys, and you're spending so much time dealing with what someone else is trying to sell them instead of being able to get your message across, which in our case I think is strong science. So that's been the most difficult, I think.
Any advice we've got a few VCs in the audience here. Any advice you'd give them for how they should treat entrepreneurs and scientists like yourself?
Well, I would say do your homework, but I know they are. I would say, I've been joking with Edgar that I've had people order DoorDash on me while I'm pitching. I would very much appreciate if that didn't happen. But I think it's the steak, not the sizzle, right? It can't just be about chasing the next shiny thing. And I think we have some real ribeye down here in Florida, some really good technology. And I think you don't have to come from Cambridge or the Bay Area to be a good scientist with excellent technology.
Absolutely. Well, Jen, thank you so much.
You're welcome.
Thank you for participating.
Of course.
And now for something completely different, we've got Sam Tarell and CJ Champion, and I'll turn it over to you, Sam. Take it away.
I think the mic up there.
Oh, okay.
Hello? Is this on? Testing? Okay, cool. How's everybody doing? Good? My name is Sam Tarell. I'm here with CJ Champion right over here with the basketball. Great. We're going to be doing something fun for you guys here at the end. So are you guys ready to have some fun? Give me a woo if you're ready. Love it. But first, I'm going to tell you a little bit about divergent thinking and how it applies to creativity and innovation, and some of the things that I've personally worked on with Hoop Brothers. This is the company that I started 10 years ago in basketball, and also Divergent U, which is a leadership academy. We work with students to help them cultivate their ideas using divergent thinking. So first, I want to explain the difference between convergent and divergent thinking. So convergent thinking is multiple variables that lead to a singular correct solution. So a simple example of that, 2 plus 2, the answer is 4. It's a convergent question with a standardized answer that's convergent thinking. A divergent question would be something like, how can we improve upon a complex system? Requires a lot of variables. That's not a simple answer. And everybody who presented today is a very high-level divergent thinker. They have gone far outside the norm and created innovations that are too complex systems to improve things. So divergent thinking is at the root of innovation. It's creative thinking, it's creativity. There was actually a case study done by Breakpoint and Beyond. They tested over 200,000 students from kindergarten through the graduation of high school. So the way they test divergent thinking is they'll ask questions like, how many uses can you think of for a paperclip? So the more uses you can think of, the higher your divergent thinking level. There's obviously infinite uses for a paperclip, but how many you can think of in a certain period of time shows your divergent thinking level. And when they tested kindergartners, 98% of the kindergartners tested at a genius level in divergent thinking. So their creativity was off the charts. They could come up with so many unique answers to a question like that. But what we see as students advance through the education system with standardized tests and a lack of divergent and creative thinking taught that only 2% of high school graduates test at the same level. So 98% of kindergartners are geniuses in divergent thinking, very creative, and only 2% of high school graduates test at that same level. Quickly, we're going to do a divergent thinking exercise. So I want everybody here to think of your favorite word. What is your favorite word? I'll give you guys maybe 30 seconds if anybody wants to shout it out. What's your favorite word? Interesting question, right? Divergent question. Could be anything. Hope. Infinity. Magic. All right, good. Hope, infinity, magic. Now, what I want you guys to do, and this is going to be a tough exercise, I hope everybody participates or at least attempts this. We're going to put 4 minutes on the clock on my phone, and you guys are going to have 4 minutes to write a 4-line poem explaining why the word you chose is your favorite word. So this is a divergent thinking exercise. It forces you to be creative. It forces you to think outside the box. And in that 4 minutes, it's a time limit. So your brain is forced not to judge, just get something on the page. So you 4 minutes to think of a poem, 4-line poem, explaining why the word you chose is your favorite word. And the words that we heard were, I think, hope, infinity, and what was the last one? Magic. Hope, infinity, and magic. So I'm actually going to do this with you guys. But I'm going to put 4 minutes on the clock starting now. Got 3 minutes left. Got a minute and a half left. All right, 10 seconds left. All right, time is up. Does anybody want to share? Anybody, any brave volunteers? Yes. You said hope, right? No, that was him. Okay, what was your word and what's your poem? Challenge. Nice. Let's hear it. Give it up. That was amazing. Any others? Anybody else want to share? Got one in the back. Your word was also challenge. Nice. Triumph. Triumph. Awesome.
Testing. Testing. There we go. Wow. So my word, once again, is triumph. We toil and fall, fail as we must. Long days, short nights, my soul alight. The music I make makes earth quake. The triumph I achieved is what I make.
Awesome. Amazing. I wrote one as well. I took the words that you guys said. So it was hope, infinity, and magic, and wrote one in 4 minutes that we had. So, have you ever felt the magic of hope? How it reaches past the infinite and inspires us to create in the unknown. A simple poem, combining words, aligning statements, deciding which lines will find the most magic. Like magicians we create on the eve of Halloween, but these aren't tricks, treats of innovation echoing through the halls of UF. I guess past the mirrors and smoke, we can use divergent thinking to find the hope. Thank you. So that's just a simple example of divergent thinking. I appreciate you guys participating. Thank you to the people that shared. It's a quick activity, but it teaches us how to turn off that judge in our mind. The judge that puts us on that point of writer's block, which is not just something writers experience, it's something business people experience too. When you have an idea that you're trying to work through and you're looking at the page and you're trying to create something, or you're trying to find a solution, sometimes you get stuck. And so divergent thinking and putting it on a timer gives you the ability to test quickly. I don't believe in writer's block. I think writer's block is perfectionism. It's saying I can't write something good, but you can write something on the page and it gives you something to work off of. And that's how you look at that from a creative perspective, but you can also apply that to business ideation. And so I applied that when I was 18 years old, starting my first business, Hoop Brothers. I grew up a basketball player. Basketball was always my passion. And One example I'll give you, Hoop Brothers creates videos for high school basketball players. We film at events all throughout the United States and produce videos to help high school basketball players get scholarships for college. They use that essentially as a commercial to promote themselves to colleges to earn scholarships. And at one event, one of the first events I did, I'm standing on a court. We have 3 videographers. There's 4 courts that we're filming. And in that moment, I experienced a lot of challenges because one of our cameras lost its SD card, filled up way earlier than I thought it would. Our battery charger broke, something fell on it. And so now we're 2 cameras down in an event, midway through an event where we have to solve this problem in the next 10 minutes. Otherwise, we're gonna have to refund players that bought videos. And so very similar to the exercise we just did, there's a time limit where I have to solve this problem. Otherwise, the relationship that we had with the vendor that we were working with at the event, we could go out of business. We could have refunds. This was our first event. So this was a massive opportunity for me. As an 18-year-old kid who knew nothing about business, I was just there filming a game and producing videos for players. And so in those 10 minutes, I had someone on my support team run to a local Walmart to pick up an SD card and a battery. I had started scouring the audience for someone with a video camera to see, could I pay this person $10, $20 to film a game so that we didn't miss that game? And so that was my beginning of entrepreneurship, was being in that intense environment, trying to find solutions. And luckily, with 2 minutes left on the clock before the game came, the support team came back with that SD card, with the battery. We got somebody from the crowd to film another game, so we were able to solve that problem. But in business and in innovation, we are put in positions in leadership where we have to make quick decisions, and that requires us to think on the fly. And so what seems like a simple exercise in a creative poem can apply in a lot of different ways to business. So CJ is actually somebody that I hired as a videographer years ago. And he started working as a videographer for Hoop Brothers, but was developing his passion in dunking. So now he's a pro dunker, and he dunks a basketball. You guys will get to see that in a couple minutes here. But divergent thinking not only applies to solving problems in business, but it also applies to leadership. So if you think about convergent, again, it's converging to a single point, multiple variables converging on one point. If you think about that in leadership, it's a leader at the top that everything is converging on. And so it's almost a ladder that people are trying to compete for and climb to reach that top spot, versus divergent leadership is a leader in the center and it's expanding out from that person. So the organization chart doesn't look like a typical organization chart where you have the CEO at the top. It's the founder in the middle, and it expands from that person and their ideas. And so I took this in a very unique, different way at Hoop Brothers and developed an entrepreneurship internship program where I started working with students to help them learn how to start their own businesses based off of what are your ideas that you're passionate about. And this wasn't very, very different from a lot of what we're talking about today with venture capital. It was much smaller projects. So a website developer, a natural soap company, to helping people come up with creative ideas and turn them into businesses through this internship program that we started at at Hoop Brothers, which then became Divergent U. So as I'm building Hoop Brothers, someone like CJ, there was opportunities for him to grow in the company and become a leader in the company, but he turned that down and decided he wanted to pursue this passion as a dunker. And so as a Divergent leader, I encouraged him to do that. Rather than saying, you have to be a part of what we're doing here, I allowed him to start this side hustle as a dunker. And he built a bit of an audience on social media doing that, and then has since developed and traveled the world as one of the top 10 professional dunkers in the world. And from that, I started Divergent University. So I hired a team to manage the day-to-day operations of Hoop Brothers. I'm still a 90% owner in Hoop Brothers, but I'm very hands-off. So my team manages the day-to-day operations of Hoop Brothers, and I've been able to focus on Divergent U. And so Divergent U is taking this concept of divergent thinking and applying it again to students. Completely self-funded the last 3 years. We have worked with over 50 student entrepreneurs to help them turn their ideas into reality. A few of the businesses that we've worked with, we had a soap company called Free the Roots. They grew 100,000 followers on Instagram. They didn't have any startup investment and got to 6 figures just building an audience organically. Again, we worked with website development agencies. I've worked with athletes. We've worked with musicians, creatives, artists, card games, very unique and creative projects that Divergent U's attracted. And it's essentially a leadership academy where we have a 3-month accelerator program working primarily with 18 to 24-year-old students, helping them turn their ideas into reality. And next fall, we're actually launching a gap year program in Orlando where we're going to be beta testing a full year curriculum with 36 students, really working on personal development. We believe that the entrepreneur needs to develop themselves, not just their business. And so some of those words that we heard, challenge, hope, infinity, triumph, and some of the words that we want to instill have to come first in the entrepreneur. And so you have to be able to build grit, you have to be able to build character, you have to be develop leadership, and that's what we do at Divergent U. So we're launching this gap year program next fall with 36 students in Orlando, where we're going to be testing these divergent thinking principles. We've written a curriculum that we are working with experts to develop even further. Gave a TEDx talk in 2019 talking about divergent thinking and divergent leadership. And we continue to advance these principles to help people build on their goals. So without further ado, I'm going to announce CJ Champion, and he's going to put on a bit of a dunk show for everybody. So if you guys can turn this way.
I also say something really quick, maybe? Just add final piece, I guess. Is this on? Testing, testing, no. I could speak loud, but whoever says I have, just to add on stuff. This one thing obviously, Switch performance would have been a little bit better, but happens sometimes. So, come off of Divergent thinking, I just want to say really quick, it does help by getting me to competitive edges. Of course, competing at the highest level, you do have to think divergently. Today would have been great with making a really interesting layup, but it's what it is. But yeah, to compete at the highest level, you do have to be creative. Come up with things that people haven't seen before. And the other biggest edge for me that I've learned recently, going through Divergent, very small in the world of basketball. You wouldn't think there's a community, but there is a really big dunking community. But making a business out of it. So with that, I've been able to take it and be able to come to speaking events like this, birthday parties, commercials, sponsorship, TV shows, travel across the country, dunk contests, and all sorts of things. So I just wanted to say that was a big help. And thank you. That's kind of all I have.